Welcome to our channel. Long before the first footprints ever touched dry land, the oceans ruled our planet. They were older than mountains, deeper than memory, and they held a world unlike anything we know today. Earth was still shaping itself, continents only just forming. And in those vast waters, life was busy experimenting. What's striking is that we actually know more about the dinosaurs who came millions of years later than we do about these forgotten oceans. So the question remains, what other monsters are still waiting, locked away in stone for us to discover? Before we begin, let us know in the comments below where in the world you're watching from and what time it is for you there. Now, settle in for a journey to the prehistoric era on Earth. Our story begins not with an animal, but with a planet. To understand the dawn of complex life, we must first understand the world that was waiting for it. We travel back in time, past the age of dinosaurs, past the first forests, to a period known as the Neo-Proterozoic Era, an immense stretch of time from 1 billion to 541 million years ago. For much of this era, the continents of Earth were not scattered as they are today. They were fused together into a single colossal land mass. Geologists call this supercontinent Rodinia, from a Russian word meaning to give birth. Rodinia dominated the globe, a vast and barren expanse of rock under a younger, fainter sun. The oceans surrounding it were immense, but the stage for animal life had not yet been properly set. Around 750 million years ago, this colossal continent began to show its age. Deep within the Earth's mantle, plumes of superheated rock pushed upwards against the crust, stretching and thinning it. Great rifts began to tear Rodinia apart. This was not a single clean break, but a long, violent, and messy continental divorce that lasted for millions of years. As the pieces of Rodinia drifted apart, they created new, smaller continents, and more importantly, they created new coastlines. The breakup of a supercontinent fundamentally changes the planet's geography. Long, continuous coastlines are replaced by intricate networks of shores, bays, and inlets. This process also created vast, shallow seas that flooded the newly formed continental shelves. These shallow, sunlit waters would become the nurseries for future life. But first, the planet would plunge into an unprecedented cold spell. This period, known as the Cryogenian, witnessed the most severe ice ages the Earth has ever known. There were at least two major pulses of glaciation, the Sturtian and the Marinoan. Evidence from glacial deposits found all over the world, even in rocks that were formed at the equator, has led to a startling hypothesis: Snowball Earth. The theory suggests that for millions of years, the entire planet, or nearly all of it, was covered in a thick sheet of ice and snow. The oceans may have been frozen over to a depth of 1 kilometer or more. This deep freeze was likely triggered by the breakup of Rodinia. The formation of new coastlines increased rainfall, which, in turn, scrubbed the greenhouse gas carbon dioxide from the atmosphere. With less carbon dioxide to trap heat, global temperatures plummeted. As ice sheets grew from the poles, their white surfaces reflected more sunlight back into space, a process called the ice albedo feedback, which cooled the planet further until it was locked in ice. Life, which at this point consisted only of single-celled microbes, faced its greatest challenge. How could anything survive on a frozen world? The answer lay in small pockets of refuge. Life likely clung on in areas around volcanic hydrothermal vents on the seafloor, which provided warmth and chemical energy. Some may have survived in tiny pools of liquid water on the ice surface, or in a hypothetical band of equatorial slush where the sun was strongest. For millions of years, this was the state of the planet, a white, silent orb seemingly hostile to life. But the same geological forces that caused the freeze would eventually break it. Throughout the snowball Earth episodes, volcanoes continued to erupt, punching through the ice and spewing gases into the atmosphere. Crucially, they released immense quantities of carbon dioxide. With the oceans sealed by ice and no rain to wash it away, this carbon dioxide built up in the atmosphere for millions of years. Eventually, it reached a tipping point where the greenhouse effect became so powerful that it overwhelmed the cooling effect of the ice. The Great Thaw began. In a geological blink of an eye, perhaps just a few thousand years, the planet transformed from an icehouse into a hothouse. Global temperatures soared. The melting glaciers unleashed torrents of fresh water into the oceans and dumped colossal amounts of sediment onto the seafloor. This process of weathering, as meltwater ground rocks into dust, released a massive pulse of nutrients, particularly phosphorus, into the marine environment. The oceans were suddenly fertilized on a planetary scale. As the Cryogenian period ended around 635 million years ago, the continents were continuing their slow dance. They briefly came together again to form a new, smaller, and more short-lived supercontinent called Pannotia. Unlike Rodinia, which was centered on the equator, much of Pannotia was clustered around the South Pole. It was a lopsided congregation of the land masses that would eventually become Laurentia, the core of North America, Baltica, Northern Europe, Siberia, and the massive supercontinent of Gondwana, which contained the future continents of Africa, South America, Australia, Antarctica, and India. The existence of Pannotia was fleeting. It too began to break apart almost as soon as it formed, at the transition from the Proterozoic Eon to the Paleozoic Era. This final breakup was the true beginning of the world we're exploring. As Pannotia rifted, it once again created immense areas of shallow, warm seas around the new, smaller continents. These epicontinental seas were the perfect environment for life. They were warm because many of the continents now straddled the equator. They were shallow, meaning sunlight could penetrate to the bottom, fueling photosynthesis. And most importantly, they were now saturated with the nutrients that had been unlocked by the melting of the snowball Earth glaciers. This nutrient-rich water fueled explosive blooms of algae and cyanobacteria. As these organisms lived and died, their organic matter sank to the sea floor. The process of decay consumed oxygen, but their photosynthetic activity produced it. Over millions of years, this biological pump began to fundamentally change the chemistry of the oceans and the atmosphere, gradually raising oxygen levels. The stage was now perfectly set. The world was warm, the oceans were shallow and nutrient-rich, and the atmosphere was slowly becoming breathable. It was a new world, ready for life to perform its greatest experiment. In the aftermath of the planet's most extreme ice ages, the newly formed shallow seas were a chemical soup brimming with potential. For over 3 billion years, life had been an exclusively microscopic affair. But now, in the final chapter of the Proterozoic Eon, something new began to appear in the fossil record. This period, lasting from roughly 635 to 541 million years ago, is known as the Ediacaran. It is named after the Ediacara Hills in South Australia, where in the 1940s, geologist Reg Sprigg first found the fossilized impressions of strange, soft-bodied organisms that defied all classification. These were the first large multicellular creatures to inhabit the Earth, and they are collectively known as the Ediacara biota. Finding them was a challenge. Unlike later animals that left behind hard shells and bones, these organisms were entirely soft. Their remains were preserved only as ghostly imprints in sandstone, fossilized snapshots of the seafloor on which they lived. Since their initial discovery, similar fossils have been unearthed across the globe in places as far-flung as the misty coasts of Newfoundland, the deserts of Namibia, and the shores of the White Sea in Russia, confirming this was a worldwide phenomenon. The creatures of the Ediacara were profoundly strange, so alien that scientists have debated their place in the tree of life for decades. They bore little resemblance to the animals of today. Many appeared to be constructed in ways that are no longer used by nature. One of the most common forms was the frond-like organism. Fossils of Charnia, for instance, look like a simple leaf or fern anchored to the deep sea floor where no light for photosynthesis could have reached. It was composed of branching fractal patterns, a simple geometric rule repeated at smaller and smaller scales to build its body. It likely absorbed nutrients directly from the seawater through its vast surface area, a lifestyle known as osmotrophy. It was utterly stationary, living and dying in one place. Perhaps the most iconic of the Ediacara organisms is Dickinsonia. These creatures were flat, oval-shaped and ribbed, looking something like a quilted bath mat. Some specimens are small, only a few millimeters across, while others reached lengths of over a meter. For a long time, scientists were unsure if Dickinsonia was a lichen, a giant single-celled amoeba, or something else entirely. The mystery began to clear with the discovery of trace fossils, fossilized tracks that showed these organisms could move. They slowly glided across the microbial mats that carpeted the seafloor, grazing as they went. The final piece of evidence came from the chemical analysis of exceptionally preserved fossils from Russia. Scientists found molecules of cholesterol, a type of fat that is a distinctive biomarker for animal life within the fossilized tissue of Dickinsonia. This was the smoking gun. Dickinsonia was not a fungus or a protist, but one of Earth's earliest animals. Other creatures from this lost world are equally perplexing. Sprigina was a segmented organism a few centimeters long with a crescent-shaped structure at one end that has been interpreted as a head. Its segmented body led some early researchers to propose it as an ancestor to the trilobites and other arthropods. But this relationship is now heavily disputed. Then there was Cambrian, a more complex organism that resembled a slug. It had a tough but flexible upper covering and is often preserved with a long proboscis extending from its body. Trace fossils show that Cambrian used this proboscis to scratch and graze on the microbial mats, indicating a more advanced feeding strategy than simple absorption. Its body plan has led many scientists to consider it a primitive relative of the mollusks, the group that includes modern snails and clams. Organisms like Phoenicia, which resembled a collection of tubes, have been found growing in dense clusters. Analysis of their growth pattern suggests they reproduce sexually by releasing sperm and eggs into the water, a strategy common in simple animals like corals and sponges. The central question surrounding the Ediacara biota is, what were they? Are we looking at the direct ancestors of the animals that would later populate the Cambrian seas? Or were they a completely separate evolutionary experiment that ultimately failed? One early hypothesis proposed that these organisms belonged to their own vanished kingdom of life, the Vendozoa, characterized by a unique quilted or fractal construction. In this view, the Ediacarans were an evolutionary dead end, completely wiped out at the end of the period, leaving no descendants. Another view holds that they represent the stem groups of modern animal phyla. A stem group animal is an early relative that possesses some, but not all, of the defining features of a modern group. In this interpretation, Cambrian could be a stem mollusk, and Dickinsonia could be a very early animal that doesn't fit neatly into any modern category, but is on the same branch of the tree of life. The current scientific consensus is more nuanced and leans towards a combination of these ideas. The Ediacara biota was likely a diverse collection of organisms. It probably included some true stem group animals, the direct ancestors of later life. It also likely included a host of strange creatures representing evolutionary experiments that went nowhere, true dead ends like the Vendozoans. And it may have even included non-animals like giant single-celled protists or fungi, all living side by side. The world they inhabited was a tranquil one, often called the Garden of Ediacara. There is very little evidence for predation. No creatures had shells, spines, or jaws. Life was largely sessile and passive. The main source of food was the thick mat of bacteria and algae that covered the seafloor. Life consisted of gently grazing on these mats or simply absorbing nutrients from the water. Then, as the Ediacara period drew to a close around 541 million years ago, these strange organisms vanished from the fossil record. Their disappearance coincides directly with the beginning of the next geological period, the Cambrian. Why they disappeared is another topic of intense debate. Perhaps their peaceful world was invaded by the first true predators, against which their soft, defenseless bodies stood no chance. Perhaps they were outcompeted for resources by the new, more active and more efficient animals of the Cambrian. Or perhaps their demise was caused by a changing environment, as shifts in ocean chemistry, possibly rising oxygen levels, favored new types of metabolisms that the Ediacara could not adapt to. It is also possible that they did not disappear entirely, but that they simply evolved into the creatures that replaced them, their forms changing so much that we no longer recognize them. Whatever the cause, their extinction cleared the stage for an evolutionary event of unparalleled speed and creativity. The quiet, strange garden was about to be replaced by a battlefield. The transition from the Ediacara period to the Cambrian period, 541 million years ago, marks the single most important evolutionary event in the history of life on Earth. The tranquil, slow-moving world of the Ediacara biota vanished, and in its place, the fossil record reveals a sudden and dramatic riot of new life forms. This event is known as the Cambrian Explosion. The term explosion is used to emphasize the geologically rapid time frame in which this diversification occurred. While it may have unfolded over a period of 10 to 25 million years, in the context of Earth's immense history, this is the blink of an eye. In this short window, nearly all of the major animal body plans that still exist today appeared for the first time. To understand the significance of this, we must understand the concept of a phylum. A phylum is one of the highest levels of classification in biology, representing a fundamental and distinct body plan. For example, arthropods, with their jointed legs and exoskeletons, are one phylum. Mollusks, with their soft bodies and shells, are another. Chordates, the phylum to which humans and all other vertebrates belong, are defined by features like a notochord. Before the Cambrian, there were perhaps only a handful of animal phyla, including the sponges, the cnidarians, jellyfish, and their relatives, and the strange creatures of the Ediacara. But in the early Cambrian, the fossil record suddenly includes the ancestors of almost everything else. We see the first arthropods, such as the iconic trilobites. We see the first brachiopods, shelled animals that would dominate the Paleozoic seafloor. We see the first echinoderms, the ancestors of starfish and sea urchins. And critically, we see the first chordates, small fish-like creatures such as Pikaia or Milocunmingia, which are our own distant relatives. It was as if the blueprints for the entire animal kingdom were drawn up and put into production all at once. This explosion of diversity was driven by the evolution of new biological hardware. One of the most important innovations was the skeleton. The very beginning of the Cambrian is marked in the rock record by the appearance of what are called small shelly fossils. These are tiny, millimeter-sized plates, spines, and tubes made of calcium carbonate and calcium phosphate. For the first time, animals were building hard parts. Skeletons provided crucial advantages. Externally, shells and sclerites offered protection from a dangerous new threat, predators. Internally, a skeleton provides a rigid structure for muscles to attach to, allowing for more powerful and precise movement. Another revolutionary innovation was vision. While simple light-sensing spots may have existed earlier, the Cambrian saw the evolution of the first true complex eyes. The compound eyes of trilobites, for instance, were remarkably advanced, made of dozens or even hundreds of individual calcite lenses. An animal that can see its environment can navigate, find food, and spot danger in ways an unseen creature never could. These new technologies fueled the rise of an entirely new ecological interaction: active predation. In the Ediacara, life was passive. In the Cambrian, life became a race. The seas were now home to the first super predator, a creature called Anomalocaris. Reaching up to a meter in length, it was a giant for its time. It had large, stalked compound eyes, a pair of spiny, grasping appendages at the front of its head for seizing prey, and a circular, tooth-lined mouth for crushing the shells of its victims. The appearance of predators like Anomalocaris kicked off a co-evolutionary arms race. If there is something that can hunt you, there is immense evolutionary pressure to develop a defense. This pressure drove the evolution of armor, spines, and burrowing behaviors in prey animals. In turn, this drove predators to evolve better senses, sharper claws, and more powerful jaws. This feedback loop, where attack and defense constantly drive each other to new levels of sophistication, dramatically accelerated the pace of evolution. The question of what caused the Cambrian explosion is one of the biggest in science, and there is no single answer. Instead, it was likely a perfect storm of environmental, ecological, and genetic factors that came together at the right time. The environmental stage was set at the end of the Proterozoic. The breakup of the supercontinent Pannosia created vast, shallow seas, and the end of the snowball Earth glaciations had flooded these seas with essential nutrients like phosphates. This fueled algal blooms, which in turn pumped oxygen into the atmosphere and oceans. For most of Earth's history, oxygen levels had been too low to support large, active animals. The metabolism required for hunting, moving quickly, and growing a large body is incredibly energy-intensive and requires a lot of oxygen. The Cambrian may have been the moment when oxygen levels finally crossed a critical threshold, making the energetic lifestyles of complex animals possible. The ecological trigger was the arms race between predator and prey. Once predation began, it created a whole new set of rules for survival. It wasn't enough to just sit and absorb nutrients anymore. Now animals need to actively seek food and avoid becoming food. This drove specialization and the rapid filling of new ecological niches. A seafloor that once only had stationary grazers now had burrowers, swimmers, hunters, and shelled defenders. Ecosystems became dramatically more complex, with energy flowing through multiple levels of a food web for the first time. Finally, there was a genetic trigger. The genetic toolkit for building complex animal bodies, controlled by regulatory genes like the Hox genes, did not magically appear in the Cambrian. Genetic evidence suggests that this toolkit had already evolved during the Ediacara period. The Hox genes act like master switches, telling different parts of an embryo where to grow a limb, a head, or a tail. The Ediacara creatures likely already possessed these genes. The explosion, therefore, may not have been caused by the evolution of new genes, but by the evolution of new ways to use those genes. Once the environmental conditions were right and the ecological pressures were in place, life had the genetic recipes on hand to start experimenting with new body plans at an unprecedented rate. The potential was already there. The Cambrian was simply when that potential was finally unleashed, filling the empty oceans with the first members of our own extended family and laying the foundation for the next 500 million years of animal evolution. In the Canadian Rocky Mountains of British Columbia lies a fossil deposit unlike any other. Known as the Burgess Shale, it is a window into a world that existed over 505 million years ago. It is not a graveyard of shells and bones, but a preserved community of soft-bodied animals buried in an instant by an ancient submarine mudslide. This rapid burial in water, with very little oxygen, prevented decay and preserved the faint impressions of creatures so strange they challenge our very definition of what an animal should look like. These are the weird wonders of the Cambrian period, life's first and perhaps most bizarre draft. For decades, scientists who studied these fossils struggled to place them into any known animal group. They were experiments in evolution, many of which belong to lineages that have long since vanished from the Earth. Among the most famous of these biological puzzles is an animal named Opabinia. When its fossil was first formally presented to a meeting of paleontologists in 1972, the audience initially laughed, believing it to be a joke. The creature was simply too alien. Opabinia had a soft, segmented body, typically about 5 to 7 centimeters in length. Along the sides of its body were lobes that likely functioned as fins, allowing it to swim through the water with an undulating motion. At the back, it had a tail fan composed of 3 pairs of upward-pointing flaps, which would have been used for steering. But it was the front of Opabinia that defied all expectations. Protruding from its head were 5 eyes perched on short stalks, giving it a 360-degree view of its surroundings. No other known animal in the history of life has possessed this precise number and arrangement of eyes. Below the eyes, where a mouth should be, there was nothing. Instead, its mouth was located underneath its head, facing backwards. To get food to this awkwardly placed mouth, Opabinia was equipped with another unique feature, a long, flexible proboscis, or nozzle, that extended from the front of its head. This proboscis, which was about 1/3 of the animal's total body length, ended in a claw-like structure armed with spines. It is believed that Opabinia would swim just above the seafloor, using its 5 eyes to search for small, soft-bodied prey in the mud. Once it located a meal, it would use its flexible nozzle to reach down, grasp the victim with its terminal claw, and then curl the entire apparatus underneath its body to deliver the food to its rear-facing mouth. The combination of these features, the 5 eyes, the vacuum cleaner-like nozzle, and the backward mouth, makes Opabinia a creature with no modern equivalent. For a long time, scientists could not figure out where it fit in the tree of life. But it is now thought to be an early ancestor of the arthropods, the group that today includes insects, spiders, and crustaceans. If Opabinia was strange, another Burgess Shale resident was, for a time, utterly incomprehensible. Its name is Hallucigenia, a name given to it because of its dreamlike and bizarre appearance. For many years, our understanding of this creature was completely wrong. The first reconstructions, based on the flattened fossils, showed an animal walking on 7 pairs of rigid, sharp spines with a row of fleshy tentacles waving uselessly on its back. No one could figure out how it could possibly walk on stiff stilts or what the tentacles were for. It also had no obvious head, with one end being a shapeless blob. It was a creature that seemed to make no functional sense, a true hallucination from the deep past. It took new fossil discoveries and a radical re-examination in the 1990s to solve the puzzle. Scientists realized they had been looking at the animal upside down and backwards. The sharp spines were not legs. They were defensive armor that pointed upwards from the animal's back, protecting it from predators. The features that were thought to be tentacles were actually its legs. Hallucigenia walked on 7 pairs of slender, claw-tipped legs, which were much better suited for movement than the stiff spines. The shapeless blob at one end was not its head, but rather fluids and decay matter squeezed out of the body during fossilization. More complete fossils found in China eventually revealed the true head at the opposite end. It was a simple, elongated structure with a pair of primitive eyes and a mouth. This tiny animal, only a few centimeters long, was a bottom dweller, likely scavenging for food particles on the seafloor. Its long legs would have elevated its body above the mud, while its formidable back spines would have made it a very unappetizing meal for any predator that tried to eat it. Modern analysis has placed hallucigenia in a group of animals called lobopodians, which are considered to be the ancestors of today's velvet worms, which live in the leaf litter of terrestrial forests. The journey to understanding this creature shows just how difficult it can be to interpret these ancient and alien body plans. Opabinia and hallucigenia were not the only oddities. The Burgess Shale is filled with creatures that represent evolutionary dead ends, or the very first steps of familiar lineages. There was Wiwaxia, a slug-like animal covered in a coat of mail made of small scale-like plates called sclerites, with 2 rows of long, sharp spines running down its back for protection. It had no eyes and a unique feeding apparatus with 2 rows of backward-pointing teeth. Then there was Morella, nicknamed the lace crab, the most common fossil in the shale. It was a small arthropod with a distinctive head shield that swept backwards into 2 long spines. It had 2 pairs of antennae and a body of numerous segments, each with a pair of feathery gills and a walking leg. These creatures and dozens of others paint a picture of a Cambrian world teeming with anatomical experimentation. Evolution was trying out countless different ways to build an animal body. The rules of life were not yet written in stone, and the result was a sea filled with weird wonders that would be unimaginable if not for their ghostly outlines preserved in the rocks of a Canadian mountain. For billions of years, life in the oceans was a largely passive affair. The dominant life forms were single-celled organisms and simple, soft-bodied creatures that floated in the water or sat on the seafloor, absorbing nutrients or filtering tiny food particles. There was competition for resources, but there was little to no active hunting. The Cambrian Explosion, which began around 541 million years ago, changed everything. This period saw the rapid appearance of most major animal groups, and with this diversification came a revolutionary new ecological interaction: active predation. Animals evolved features specifically for hunting other animals, such as sharp claws, complex eyes, and powerful jaws. This development created a powerful new force in evolution, an arms race between predator and prey that has shaped life ever since. The first great hunter to emerge from this new world, the undisputed apex predator of the Cambrian seas, was a creature whose name means abnormal shrimp, Anomalocaris. The story of how we came to know Anomalocaris is a classic tale of paleontological detective work. For over a century, its different body parts were discovered separately and were so unusual that they were identified as 3 completely different animals. The first parts to be found were its 2 large, spiny frontal appendages. These were thought to be the bodies of a shrimp-like crustacean, which is how the name abnormal shrimp came about. Another common fossil found in the same rock layers was a circular, ring-like structure that looked like a flattened jellyfish. It was given the name Paleozoic. Finally, the main body of the animal, a large, soft, segmented form without any appendages, was thought to be a type of sea cucumber or another primitive, slow-moving creature. No one suspected that these 3 very different fossils, the shrimp, the jellyfish, and the sea cucumber, were actually all parts of one giant animal. The mystery was finally solved in the early 1980s when a scientist was clearing rock from a large, complete fossil slab from the Burgess Shale. He found one of the shrimp appendages in a circular jellyfish mouth attached to the front of the same animal. The puzzle pieces snapped into place, revealing the true nature of Anomalocaris. It was not a small shrimp, but a huge, swimming predator, the largest and most formidable animal of its time. Fully assembled, Anomalocaris was a fearsome creature. It could reach lengths of up to 1 meter, possibly even 2, making it a giant in a world where most other animals were only a few centimeters long. It was an active swimmer, propelled through the water by a series of flexible lobes that ran down the sides of its body. By undulating these lobes in a wave-like motion, it could move with speed and agility far more effectively than the clumsy swimmers of earlier times. At the back of its body, a tail fan helped it to steer. At the front of its head, it possessed two large compound eyes mounted on stalks. These were among the most advanced eyes of the Cambrian period, each containing thousands of individual lenses. This would have given Anomalocaris excellent vision, allowing it to spot prey from a distance. But its most terrifying features were its weapons. Protruding from the front of its head were the two appendages that had once been mistaken for shrimp. These powerful, segmented arms were lined with sharp, durable spines and could be used to grasp and spear its prey. Once captured, the victim would be brought towards the animal's mouth. The mouth of Anomalocaris, the part once called Paleozoic, was a circular structure made of 36 hard, overlapping plates. These plates had sharp, tooth-like serrations on their inner edges. It is believed that the mouth could not close completely flat, but it could constrict like the aperture of a camera, creating a powerful biting and crushing mechanism. This unique mouth was perfectly suited for breaking through the hard exoskeletons of its preferred prey. The primary target for Anomalocaris was likely the most common armored animal in the Cambrian seas, the trilobite. The evolution of this super predator created immense pressure on other animals to develop defenses. The hard shells, spines, and burrowing behaviors of many Cambrian creatures were likely direct responses to the threat posed by hunters like Anomalocaris. Fossil evidence supports this predator-prey relationship. Paleontologists have found many trilobite fossils with distinctive W-shaped bite marks and injuries, particularly on their head and tail shields. For a long time, these wounds were a mystery, but the size and shape of the marks match the mouthparts of Anomalocaris. It appears that Anomalocaris would grab a trilobite with its frontal claws and then use its circular mouth to crush or puncture the exoskeleton to get at the soft tissues inside. Some fossilized droppings, known as coprolites, from the Cambrian period are packed with the crushed-up fragments of trilobite shells, further evidence of this intense predation. As the top predator, Anomalocaris played a crucial role in shaping its ecosystem. It kept prey populations in check and drove the evolutionary arms race that led to more complex defenses and behaviors. Its existence marks a fundamental shift in the nature of life on Earth, from a world of passive grazers and filter feeders to a dynamic and dangerous environment where the hunt was on. It was the first, but certainly not the last, great predator to rule the oceans. Of all the creatures that lived during the Paleozoic era, none are more famous or more emblematic of the ancient oceans than the trilobites. These remarkable animals were not a single species, but an entire class of extinct marine arthropods, a group that today includes insects, spiders, and crabs. They first appeared in the fossil record during the early Cambrian period, around 521 million years ago, and they were an immediate evolutionary success. For nearly 300 million years, they thrived in the world's oceans, diversifying into more than 20,000 known species before finally disappearing in the mass extinction event at the end of the Permian period. Their name, which means three lobes, is derived from the three distinct longitudinal lobes that run the length of their body, a central raised axial lobe flanked on either side by a pleural lobe. This tripartite body plan is the defining characteristic of the entire group. The reason we know so much about them is their hard, mineralized exoskeleton, which fossilized with exceptional frequency, leaving behind a rich and detailed record of their long history on Earth. The body of a trilobite was divided into three main sections from front to back: the cephalon, or head, a segmented thorax, or body, and a pygidium, or tail shield. The cephalon was a single fused plate that housed the stomach, heart, and the front of the nervous system. It also contained the trilobites' most sophisticated sensory organs. Most trilobites had a pair of complex compound eyes, making them some of the very first animals to see their world in detail. Uniquely, the lenses of trilobite eyes were not made of organic protein like the eyes of most animals, but of calcite, the same mineral that made up their shell. This is a feature not seen in any other creature. There were two primary types of eyes. The more common type was the hollow crawl eye, which consisted of thousands of tiny calcite lenses packed tightly together under a single corneal layer. This would have given the trilobite an incredibly wide field of view, excellent for detecting movement. The second, more advanced type was the schizocrol eye, found only in one suborder of trilobites. In these eyes, each lens was much larger, biconvex, and was separated from its neighbors, with each lens having its own individual cornea. This sophisticated optical design corrected for aberrations and likely provided stereoscopic vision, giving these trilobites excellent depth perception, a key advantage for both hunting and avoiding predators. Some trilobites that lived in the dark depths of the ocean or burrowed in mud lost their eyes entirely. The thorax was the middle section of the body, composed of a series of articulated segments. Each segment could move relative to its neighbors, giving the trilobite's body a great deal of flexibility. This flexibility allowed many species to enroll or roll up into a ball, much like a modern pill bug. This was a highly effective defensive posture, protecting their soft, vulnerable underside and antennae with their hard dorsal shell. The number of thoracic segments varied widely between species, from as few as 2 to over 100. Attached to the underside of each segment was a pair of jointed legs. These legs were biramous, meaning they had 2 branches. One branch was a sturdy walking leg, while the other was a delicate feathery gill structure used for respiration. The final section of the body was the pygidium, or tail. This was formed from a number of fused segments and, like the cephalon, was a solid plate. The size and shape of the pygidium varied enormously and is a key feature used to classify different trilobite species. In some it was tiny, while in others it was as large as or even larger than the head. Trilobites were not just one type of animal. They were incredibly diverse and occupied nearly every available ecological niche in the Paleozoic seas. Some were active predators, hunting worms and other small invertebrates on the seafloor. Others were scavengers, feeding on dead organic matter. Many were detritivores, sifting through the sediment and consuming nutrient-rich mud. Some streamlined forms were likely active swimmers in the open water column, while others were filter feeders. Their immense variety in body shape and size reflects this ecological diversity. Some were smooth and flat, adapted for burrowing. Others were covered in elaborate spines, which could have been used for defense, for stabilization on a soft seafloor, or as sensory organs. Their incredible success can be attributed to several factors. Their hard exoskeleton provided excellent protection. Their advanced eyes gave them a significant sensory advantage, and their segmented body plan proved to be highly adaptable, allowing them to evolve into a vast array of forms suited for different lifestyles. Like all arthropods, trilobites had to molt their exoskeleton in order to grow. They would split their head shield along preformed sutures, crawl out, and then hide while their new, larger shell hardened. These discarded molts are extremely common in the fossil record, even more common than the fossils of the animals themselves, and they provide an invaluable record of the life and growth of these ancient mariners. The Ordovician period, lasting from approximately 485 million to 444 million years ago, saw an unprecedented explosion of marine life known as the Great Ordovician Biodiversification Event, or GOBE. This event was a significant turning point in Earth's history, transforming relatively simple Cambrian ecosystems into far more complex and crowded marine environments. During the Cambrian, life was primarily dominated by trilobites, archaeocyathids, and primitive arthropods. While diverse, these communities were less structured and had fewer distinct ecological roles. The Gobi marked a dramatic increase in the number of marine species and families, fundamentally changing the structure of ocean food webs. Before the Ordovician, the oceans were inhabited by organisms that largely grazed on microbial mats or were detritivores, feeding on dead organic matter. Predators existed, such as Anomalocaris, but their diversity and impact were limited. The Gobi introduced a wider array of feeding strategies and lifestyles. New groups of organisms evolved, and existing groups diversified extensively. This led to a fourfold increase in marine family numbers over a period of about 20 to 25 million years. This expansion was not just an increase in numbers, but a rise in ecological complexity. Organisms began to occupy more specialized niches, leading to more intricate food chains and competitive interactions. One of the most notable developments during the Gobi was the diversification of sessile or stationary suspension feeders. These creatures, which filter food particles from the water, became far more abundant and varied. Bryozoans, for example, appeared for the first time as an important component of seafloor communities. These tiny colonial animals built intricate structures, often encrusting other organisms or forming delicate branching colonies. Brachiopods, shelled invertebrates that superficially resemble clams but are distinct in their internal anatomy, also underwent massive diversification. They became extremely abundant, forming dense beds on the seafloor. Crinoids, often called sea lilies, expanded significantly, creating vast underwater meadows. These echinoderms, related to modern starfish, attached themselves to the seafloor by a stalk and filtered food with feathery arms. Their fossil remains are common in Ordovician rocks, indicating their widespread success. The increase in suspension feeders also contributed to the establishment of the first true reef systems, as described in the next section. These bio-built structures provided complex, three-dimensional habitats, offering shelter and new feeding opportunities for other organisms. The presence of these large, stable structures further enhanced the crowding and complexity of the Ordovician seas. Mobile organisms also diversified during the Gobi. Gastropods, or snails, became more varied, adapting to different environments and food sources. Bivalves, like clams, also increased in diversity, burrowing into sediments or attaching to hard surfaces. Echinoderms, in addition to crinoids, diversified to include more varied forms of starfish, sea urchins, and other relatives. Trilobites, though already present in the Cambrian, continued to diversify, adapting new forms and ecological roles, though their overall dominance began to wane as other groups rose. Predators also evolved to meet the demands of a more crowded and diverse prey field. Cephalopods, such as the orthoconic nautiloids, became significant predators. These creatures, discussed in detail in the giant squid section, grew to impressive sizes and actively hunted other marine invertebrates. The evolution of more efficient predators likely drove an arms race in prey species, leading to improved defenses such as thicker shells, spines, and burrowing behaviors. This co-evolutionary dynamic further contributed to the complexity of the ecosystem. The causes of the Great Ordovician biodiversification event are thought to be multifaceted. One factor may have been a rise in global sea levels, leading to the expansion of shallow marine environments on continental shelves. These shallow seas, bathed in sunlight and rich in nutrients, provided vast areas for marine life to colonize and diversify. Tectonic activity, such as continental drift and volcanic outgassing, might have influenced ocean chemistry and nutrient availability, creating conditions favorable for life's expansion. Changes in ocean chemistry itself could have played a role. A possible increase in oxygen levels in the oceans might have supported larger and more metabolically demanding organisms. The development of deeper burrowing by marine invertebrates could have also oxygenated seafloor sediments, creating new habitats and promoting diversification. The increased mixing of ocean waters, driven by global climate patterns, could have brought more nutrients from deep waters to the surface, fueling primary productivity and thus supporting a larger biomass. Furthermore, the fragmentation of the supercontinent Gondwana during the Ordovician might have created more distinct oceanic basins and continental shelves, leading to geographic isolation and the evolution of unique species in different regions. This provincialism could have contributed to the overall increase in global biodiversity. The interaction of these physical and chemical changes with biological innovation, such as the evolution of jaws in fish, created a positive feedback loop that propelled the diversification. The globe did not happen uniformly across all groups or regions. Different groups diversified at different rates and times. However, the cumulative effect was a complete overhaul of marine ecosystems. The Ordovician oceans were bustling with life, far more so than the preceding Cambrian. Imagine vast seafloors covered with dense thickets of crinoids and bryozoans, interspersed with beds of brachiopods, while trilobites scuttled across the sediment and large nautiloids patrolled the waters above. This crowded, complex marine world laid the foundation for all subsequent marine life, including the vertebrates that would eventually colonize land. It demonstrated Earth's capacity for life to not just survive, but to explode in diversity when conditions are right. This crowded sea was a dynamic laboratory of evolution, showcasing the intricate dance between organisms and their environment on a planetary scale. The concept of a reef often conjures images of vibrant, colorful modern coral reefs teeming with fish in tropical waters. However, the very first major reef systems in Earth's history, which emerged and flourished during the Ordovician and Silurian periods, were built by very different organisms. These ancient structures, unlike modern coral reefs, were primarily constructed by strange sponges, known as stromatoporoids and early primitive corals, alongside other scaffolding organisms like tabulate and rugose corals and bryozoans. These pioneering reefs represent a critical step in the evolution of complex marine ecosystems, creating new habitats and significantly increasing the three-dimensional complexity of the seafloor. Before the Ordovician, during the Cambrian period, there were structures sometimes referred to as reefs, but these were typically small, discontinuous mounds built by archaeocyathids, a group of extinct cup-shaped organisms, possibly related to sponges. These Cambrian reefs were relatively simple and short-lived. The Ordovician marked the advent of true, expansive reef systems that could withstand strong currents and waves, providing stable, long-lasting habitats. The primary architects of these early Ordovician reefs were stromatoporoids. These were not the soft, porous sponges we typically imagine today. Instead, stromatoporoids were sessile, colonial organisms that precipitated robust skeletons of calcium carbonate, often forming layered, dome-shaped or columnar structures. They grew by adding successive layers of calcareous material, creating solid, wave-resistant frameworks. Their exact biological classification has been debated, but they are generally considered a group of extinct sponges or a closely related group of animals. Their ability to build sturdy skeletons allowed them to create the foundational structure for the first true reefs. Alongside stromatoporoids, early forms of corals also played a significant role. These included tabulate corals and rugose corals. Tabulate corals formed colonial structures, often looking like honeycombs or chains of tiny tubes. They built their skeletons from calcium carbonate and grew by budding, creating large, interconnected colonies. Rugose corals, often called horn corals due to their distinctive horn-like shape in solitary forms, could be solitary or colonial. Both tabulate and rugose corals were different from modern scleractinian corals, which evolved much later. These primitive corals did not typically have the symbiotic algae that modern corals rely on for much of their energy. Instead, they likely captured plankton from the water. Despite this difference, their ability to secrete calcium carbonate skeletons was crucial for reef construction. Other organisms also contributed to the construction and stability of these early reefs. Calcareous algae, which also precipitated calcium carbonate, helped to bind the reef framework together. Bryozoans, tiny colonial animals that built intricate calcified colonies, encrusted the reef surfaces and filled in spaces, adding to the structural integrity. Crinoids, with their stalked bodies, often grew abundantly on the reef surface, adding to the biomass and contributing to the filtering of water. Brachiopods, mollusks, and trilobites found shelter within the nooks and crannies of the reef structure. The formation of these first reefs was a complex ecological process. It involved primary framework builders like stromatoporoids and corals laying down the initial structure. Then, various encrusting organisms and sediment-trapping organisms helped to solidify and expand the reef. The internal spaces within the reef framework provided refugia for a variety of smaller invertebrates, protecting them from predators and strong currents. These protected microhabitats fostered an even greater diversity of life than the surrounding open sea floor. The presence of these large, three-dimensional structures had a profound impact on the marine environment. Reefs act as natural barriers, dissipating wave energy and creating calmer waters on their leeward side. This allowed for the accumulation of fine sediments and the establishment of different types of communities in the sheltered lagoons. They also influenced water currents and nutrient distribution, creating productive microclimates within the ocean. Reefs are also centers of high biodiversity. They provide numerous ecological niches, surfaces for attachment, crevices for hiding, elevated positions for filter feeding, and diverse food sources. This complexity allowed for a greater variety of species to coexist than in flat, uniform environments. The first reefs thus became bustling underwater cities, supporting intricate food webs that included grazers, filter feeders, detritivores, and a growing number of predators. Throughout the Ordovician and into the Silurian period, approximately 444 million to 419 million years ago, these stromatoporoid tabulate rugose coral reefs became widespread. They reached their peak development in the Devonian, forming massive structures that spanned hundreds of kilometers in some areas. These ancient reefs left behind significant fossil records, providing scientists with invaluable insights into early marine ecosystems. The success of these early reef builders continued until the late Devonian extinction events, which severely impacted many of these groups. However, their legacy is immense. They demonstrated the power of biological construction to fundamentally alter ocean environments, paving the way for future reef-building organisms and supporting the ongoing diversification of marine life. These forgotten structures, built by unfamiliar creatures, were among the most dynamic and biologically important habitats in the Paleozoic oceans. The term giant squid typically refers to the colossal modern cephalopods of the deep ocean, but in the Ordovician seas, a different kind of giant ruled. During this period, from approximately 485 million to 444 million years ago, the ocean saw the rise of the cephalopods, particularly the giant straight-shelled nautiloids. These ancient marine predators were unlike anything alive today, and they represented the pinnacle of the marine food web. Cephalopods are a group of mollusks that includes modern octopuses, squids, and cuttlefish, as well as nautiluses. In the Ordovician, the dominant forms were nautiloids, characterized by an external shell that was often long and conical, resembling a straight horn or tusk. While some modern nautiluses have coiled shells, many of their ancient relatives, known as orthoconic nautiloids, had straight shells. Among these, genera like Camaraoceras stood out for their immense size. While estimates vary due to incomplete fossil records, some Camaraoceras individuals are believed to have reached lengths of up to 11 meters, or 36 feet, including their shell. This made them among the largest animals on Earth during the Ordovician period, truly the giants of their time. These massive nautiloids moved through the water using a sophisticated form of jet propulsion, similar to modern cephalopods. They had a muscular mantle, or body wall, that could rapidly expel water through a funnel-like opening called a hypernome. By controlling the direction of this water jet, they could move forward, backward, or hover. This efficient mode of locomotion allowed them to be agile predators, capable of pursuing prey in the open water. Their shells were divided into a series of chambers separated by internal walls called septa. These chambers were filled with gas and liquid, which the animal could regulate to control its buoyancy. This allowed them to move effortlessly through the water column, ascending to hunt near the surface or descending to deeper waters. The head of an orthoconic nautiloid emerged from the opening of the shell. It possessed a ring of tentacles, which were likely unsuckered, unlike modern squid and octopus tentacles. These tentacles would have been used to grasp and manipulate prey. Inside the mouth, they had a powerful beak-like jaw, similar to a parrot's beak, capable of crushing the shells of armored prey. They also possessed a radula, a ribbon-like structure with rows of teeth used to rasp and process food. As top predators, these giant nautiloids preyed on a wide range of marine invertebrates. Their diet likely included trilobites, smaller nautiloids, brachiopods, and other shelled creatures abundant in the Ordovician seas. Their large size and powerful beaks made them formidable hunters, placing them at the apex of the Ordovician food web. The presence of such large predators would have exerted significant selective pressure on their prey, driving the evolution of defensive adaptations like thicker shells, spines, and faster escape mechanisms in other marine organisms. This predator-prey dynamic was a crucial element in the increasing complexity and crowdedness of Ordovician ecosystems. The straight-shelled nautiloids were incredibly successful throughout the Ordovician and into the Devonian period. Their fossils are found globally, indicating their widespread distribution in the ancient oceans. They inhabited various marine environments, from shallow shelf seas to deeper waters. Their elongated, streamlined shells would have made them efficient swimmers, while their buoyancy control allowed them to navigate different depths. While the exact lifespan and reproductive strategies of these ancient giants are unknown, it is likely they reproduced by laying eggs, similar to modern cephalopods. Their long shells would have provided protection from attack, while their jet propulsion allowed for escape. The evolution of such large and specialized predators was a testament to the increasing availability of diverse food sources in the Ordovician oceans, fueled by the Great Ordovician Biodiversification Event. The reign of the giant orthoconic nautiloids continued for tens of millions of years. However, their dominance eventually waned. While some straight-shelled forms persisted into later geological periods, they faced increasing competition from new groups of marine predators, particularly fish. The evolution of jawed fish, which became highly diverse in the Devonian period, introduced faster, more agile, and often armored competitors and predators. Over time, the coiled-shelled nautiloids, similar to the modern nautilus, became more prevalent, as a coiled shell offers better maneuverability and is less cumbersome for smaller individuals. The legacy of these Ordovician giants is significant. They represent an early example of large, active and intelligent, for their time, marine predators. Their existence demonstrates the incredible evolutionary pathways taken by early life, leading to forms that are both alien and awe-inspiring when compared to modern creatures. The fossilized shells of Cambrian and its relatives stand as silent monuments to a time when the oceans were ruled by shelled, jet-propelled giants, a true giant squid of the ancient Paleozoic world. The Eurypterids, commonly known as sea scorpions, were a diverse group of arthropods that lived in ancient waters. They first appeared in the Ordovician period, approximately 470 million years ago. Early species were relatively small, often measuring only a few centimeters in length. These creatures were part of a larger group of animals called chelicerates, which also includes modern spiders, scorpions, and horseshoe crabs. Unlike their terrestrial scorpion relatives, Eurypterids were primarily aquatic, though some later forms may have been able to spend short periods on land. Their fossil record is extensive, providing much information about their anatomy and lifestyle. The body of a Eurypterid was divided into two main parts: the prosoma, or head section, and the opisthosoma, or abdomen. The prosoma typically bore several pairs of appendages. The first pair, known as chelicerae, were small, pincer-like claws used for grasping food. These were followed by 4 pairs of walking legs, which were often spiny or bristly and used for moving across the seafloor. The final pair of appendages on the prosoma were often modified into broad, paddle-like structures. These paddles were crucial for swimming, allowing the Eurypterid to propel itself through the water. Some species had very large, powerful paddles, indicating a more active swimming lifestyle. The opisthosoma, or abdomen, was segmented and typically consisted of 12 segments. The first few segments contained the gills, which were necessary for breathing underwater. The exact structure of the gills varied among different species, but they generally consisted of plate-like structures that allowed for the exchange of gases. The final segment of the opisthosoma was the telson, or tail. The telson varied greatly in shape and size among different Eurypterid species. In some, it was a long, slender spike, while in others it was broad and flattened, resembling a rudder. Some Eurypterids, particularly the larger, later species, had a telson that resembled the stinging tail of a modern scorpion, though it is not clear if these were used for injecting venom. The exoskeleton of Eurypterids was made of chitin, a tough, protective material. This outer shell provided support and protection, much like the shells of modern crabs or insects. As Eurypterids grew, they had to shed their exoskeletons in a process called molting. Similar to other arthropods, fossilized molts are common, indicating that this was a regular part of their life cycle. The presence of molts also helps scientists understand the growth stages of these ancient creatures. The surface of the exoskeleton was often textured with small bumps, ridges, or spines, which may have provided additional protection or aided in camouflage. Early Eurypterids were likely scavengers or small predators, feeding on detritus or other small invertebrates. Their small chelicerae were suitable for picking up small pieces of food or grasping soft-bodied prey. The walking legs, with their spines, could have helped to manipulate food towards the mouth. Their relatively small size meant they were not the apex predators of their time, but rather occupied a niche lower down in the food chain. They lived in marine environments, predominantly in shallow coastal waters, estuaries and lagoons. Some fossil evidence suggests they could tolerate brackish or even freshwater conditions, indicating a degree of adaptability to different salinities. The eyes of Eurypterids were typically compound, similar to those of insects. They had 2 large, prominent compound eyes located on the top of the prosoma, providing a wide field of vision. In addition, many species possess 2 smaller, simple eyes, or ocelli, located between the compound eyes. These simple eyes likely detected changes in light intensity, while the compound eyes provided more detailed vision, helping them to navigate and locate food or avoid predators. The position of their eyes suggests they were primarily bottom dwellers, looking upwards for potential threats or opportunities. Reproduction in eurypterids is not fully understood, but it is believed they reproduced sexually, laying eggs. Some fossils show evidence of different sizes and shapes among individuals of the same species, which might indicate sexual dimorphism, where males and females had slightly different appearances. However, definitive evidence of their reproductive strategies, such as mating rituals or parental care, is scarce in the fossil record. Their life cycle likely involved several larval stages, similar to other arthropods, before reaching their adult form. The early eurypterids were not yet the fearsome sea scorpions that would dominate later periods. They were part of a diverse community of early marine life, coexisting with trilobites, early mollusks, and other invertebrates. Their evolution was a gradual process, with successive species developing larger sizes, more specialized appendages, and more predatory lifestyles. The foundation for their later success was laid during these early stages, as they perfected their basic body plan and adapted to various aquatic environments. Their presence in the Ordovician marks the beginning of a long and successful lineage that would eventually produce some of the largest arthropods to ever live. The sea scorpion's early forms were important for understanding their evolutionary trajectory. They show the basic blueprint from which more specialized and larger forms would emerge. Their segmented bodies, jointed appendages, and chitinous exoskeletons were key features that allowed them to thrive in the ancient oceans. The development of paddle-like legs for swimming was a significant adaptation, allowing them to move beyond just crawling on the seafloor. This mobility would become a defining characteristic of many eurypterid species. The presence of chelicerae, even in their small, early forms, indicates their predatory or scavenging nature from the very beginning of their evolutionary history. These early species were crucial stepping stones in the development of a group that would eventually include some of the most formidable invertebrates of the Paleozoic era. The first vertebrates to appear on Earth were the ostracoderms, a group of ancient jawless fish. These pioneering creatures emerged in the early Ordovician period, approximately 480 million years ago. Unlike modern fish, ostracoderms lacked jaws, meaning they could not bite or chew their food. Instead, they fed by sucking small particles of food from the seafloor. Their bodies were often covered in bony plates, providing a strong defense against predators in the ancient oceans. These early fish were a significant evolutionary step, representing the very first animals with a backbone or vertebral column, even if it was made of cartilage rather than bone in many early forms. A prime example of an early ostracoderm is Arundaspis, a fish that lived around 470 million years ago. Arundaspis was a relatively small fish, typically measuring about 15 centimeters in length. Its body was streamlined and flattened, well suited for life on the seafloor. The most striking feature of Arundaspis, and indeed many ostracoderms, was its extensive armor. Its head and the front part of its body were encased in large, rigid, bony plates. These plates formed a protective shield covering the brain, gills, and vital organs. The rest of its body, towards the tail, was covered in smaller, overlapping scales or plates, allowing for some flexibility. The mouth of Arundaspis was a simple, slit-like opening located on the underside of its head. Without jaws, it could not actively grasp or tear food. Instead, it is likely fed by filtering small organic particles and microorganisms from the sediment. This feeding strategy involved sucking in water and mud, then filtering out the edible bits through its gill slits. This method of feeding, known as suspension feeding or deposit feeding, was common among early marine invertebrates and was adopted by these first vertebrates. The constant intake of water also facilitated gas exchange through the gills, which were located in pouches along the sides of the head shield. Arandaspis lacked paired fins, which are characteristic of most modern fish and provide stability and maneuverability. It possessed only a simple tail fin, which would have provided some propulsion for swimming. Its flattened body and lack of strong fins suggest that Arandaspis was not a fast or agile swimmer. It likely spent most of its time resting on or slowly moving across the seafloor, using its tail for short bursts of movement to escape danger or find new feeding grounds. Its heavy armor would have made sustained swimming difficult. The bony plates of ostracoderms like Arandaspis were not just for defense. They also represent the earliest known examples of bone in vertebrates. This development of internal skeletal structures was a crucial innovation. The bone was not necessarily true bone, as we know it in mammals, but a form of dermal bone, meaning it developed in the skin, similar to the scales of some modern fish or the plates of turtles. This armor provided protection from the large invertebrate predators of the Ordovician, such as the massive nautiloids and early eurypterids. Without jaws or strong swimming abilities, defense was paramount for survival. The eyes of Arandaspis were located on the top of its head, suggesting it primarily looked upwards. This positioning would have been useful for detecting predators swimming above it. It is believed that Arandaspis had a simple, light-sensitive pineal eye on the top of its head, in addition to its two main eyes. This third eye could detect changes in light, helping the fish to sense day and night cycles or the approach of shadows from above. Ostracoderms lived in marine environments, typically in shallow coastal waters. Their fossil remains have been found in ancient shallow sea deposits, indicating they preferred these environments where food particles were abundant and the seafloor was accessible for their feeding method. They were part of a diverse ecosystem that included trilobites, brachiopods, and other early invertebrates. Their appearance marked the beginning of the vertebrate lineage, setting the stage for the evolution of all subsequent fish, amphibians, reptiles, birds, and mammals. The success of ostracoderms, despite their simple anatomy, lay in their adaptations to their specific niche. Their armor provided protection, and their filter feeding allowed them to exploit a food source that was plentiful on the seafloor. While they were not the dominant predators, they were successful survivors in a challenging environment. Their existence for millions of years demonstrates the effectiveness of their evolutionary strategy. The bony armor, initially a defensive adaptation, would later become the foundation for the internal skeletons of more advanced vertebrates. The jawless fish represent a critical juncture in the history of life. They were the first to possess a notochord, a flexible rod that supported their body, which is a precursor to the vertebral column. This innovation provided a new level of structural support and flexibility, allowing for more complex movements than seen in earlier invertebrates. While primitive by modern standards, these armored, jawless fish laid the groundwork for the incredible diversity of vertebrates that would follow. Their story is one of simple beginnings, foundational adaptations, and the first tentative steps towards the complex animal life we see today. The end of the Ordovician period, approximately 445 million years ago, was marked by the first major mass extinction event in Earth's history. This catastrophic event, often referred to as the Ordovician-Silurian extinction, led to the disappearance of more than 85% of all marine species. The primary cause of this widespread devastation was a sudden and severe global cooling, resulting in a dramatic ice age. This great freeze had profound and lasting impacts on the planet's oceans and the life within them. Before the extinction, the Earth's climate was generally warm and stable, with high sea levels. Vast, shallow seas covered much of the continents, providing extensive habitats for a diverse array of marine life. Trilobites, brachiopods, bryozoans, corals, and early jawless fish thrived in these conditions. The oceans were teeming with life, and new species were constantly evolving and diversifying. However, this period of stability was abruptly interrupted by a series of events that plunged the planet into an ice age. The onset of the Ice Age is believed to have been triggered by a combination of factors, including changes in atmospheric carbon dioxide levels and continental drift. As the supercontinent Gondwana drifted towards the South Pole, it positioned itself over the pole, creating conditions favorable for the formation of large ice sheets. The growth of these massive glaciers locked up vast quantities of water, leading to a significant drop in global sea levels. This sea level fall, estimated to be as much as 100 meters, had devastating consequences for the shallow marine ecosystems. The retreat of the seas destroyed extensive coastal habitats, including the vast shallow water shelves where most marine life resided. Organisms that relied on these warm, sunlit waters for food and reproduction found their homes shrinking rapidly. Many species were unable to adapt to the sudden loss of their preferred environments, leading to widespread population crashes. The reduction in habitat space also increased competition among the surviving species, further stressing the ecosystems. In addition to the loss of habitat, the global cooling caused a dramatic drop in ocean temperatures. Many marine organisms are highly sensitive to temperature changes and could not survive in the colder waters. Tropical and subtropical species, which were abundant in the Ordovician seas, were particularly vulnerable. The cold, deep waters, which were now able to circulate more freely, also brought up oxygen-poor water to the surface in some areas, creating dead zones where life could not be sustained. This lack of oxygen further exacerbated the crisis for marine life. The impact of the Great Freeze was not uniform across all groups of organisms. Some groups, like the graptolites, which were colonial animals that floated in the water, were severely affected. Many species of trilobites, particularly those adapted to specific shallow water environments, also suffered massive declines. Brachiopods, a type of shelled invertebrate, experienced significant losses, though some lineages managed to survive. The early jawless fish, the ostracoderms, also saw a reduction in their diversity, though they did not go extinct entirely. The extinction event occurred in two main pulses. The first pulse was associated with the initial cooling and glaciation, leading to the first wave of extinctions as sea levels dropped and temperatures plummeted. The second pulse occurred as the glaciers began to melt and sea levels rose again, leading to further environmental instability, including changes in ocean chemistry and oxygen levels. This two-phase extinction made it particularly devastating as life struggled to recover from one crisis only to be hit by another. The fossil record clearly shows the sharp decline in biodiversity at the end of the Ordovician. Sedimentary layers from this period contain significantly fewer fossils and a much lower diversity of species compared to the layers immediately preceding them. This geological evidence provides direct proof of the severity of the extinction event. Scientists study these fossil assemblages to understand which groups were most affected and how the ecosystems changed in response to the environmental pressures. Despite the immense loss of life, the Ordovician-Silurian extinction also created opportunities for new forms of life to emerge and diversify in the subsequent Silurian period. The survivors of the Great Freeze were those species that were more adaptable to changing conditions, or those that lived in deeper waters or more stable environments. These surviving lineages would go on to repopulate the oceans and give rise to new forms, eventually leading to the incredible diversity of fish and other marine life that would characterize the Devonian period. The Great Freeze stands as a stark reminder of the profound impact that global climate change can have on life on Earth. The close of the Ordovician period, approximately 443 million years ago, witnessed one of the most severe extinction events in Earth's history. Often referred to as the Ordovician-Silurian extinction, this catastrophic event dramatically reshaped marine ecosystems, wiping out a significant portion of the life that had flourished in the preceding era. Scientists estimate that around 85% of all marine species perished, making it the second largest extinction event in terms of the percentage of genera lost. The primary culprits are believed to be a combination of intense global cooling, leading to widespread glaciation and a subsequent rapid sea level fall. As vast ice sheets formed, locking up enormous quantities of water, shallow marine habitats, which were teeming with life, disappeared or became inhospitable. The subsequent melting of these glaciers then caused a rapid sea level rise and changes in ocean chemistry, including oxygen depletion, further stressing the surviving organisms. The world that emerged from this cataclysm was a vastly different place. But life, with its inherent resilience, began the slow and arduous process of recovery and diversification during the ensuing Silurian period. The survivors of the Ordovician extinction were a testament to the adaptability of life. Certain groups, though severely depleted, managed to cling on and eventually rebound. Among the most prominent survivors were some lineages of trilobites, the iconic armored arthropods of the early Paleozoic. While many families and genera vanished, a few hardy lines persisted, demonstrating a remarkable capacity to adapt to the changed environmental conditions. These surviving trilobites, often smaller and more generalized in form than their Ordovician predecessors, found new niches in the recovering oceans. Similarly, brachiopods, clam-like filter feeders, also suffered heavy losses, but some groups endured and their populations began to expand again, becoming important components of the Silurian seabed communities. Crinoids, often called sea lilies due to their plant-like appearance, also saw a resurgence. These stalked echinoderms, which filter-fed from the water column, formed extensive meadows on the seafloor, contributing significantly to the biomass and creating complex habitats for other creatures. The recovery was not merely a return to the old ways. It was a period of profound evolutionary innovation. The ecological vacancies left by the extinct species created opportunities for the survivors to diversify and for new forms to emerge. One of the most significant aspects of this rebound was the continued evolution of fish. While jawless fish, or ostracoderms, had appeared in the Ordovician, they truly began to proliferate and diversify in the Silurian. These early fish were armored with bony plates, providing protection against predators, and they were primarily bottom dwellers, sifting through sediments for food. Their forms became more varied, with different body shapes and fin arrangements, indicating an exploration of various ecological roles. This period also saw the development of more sophisticated sensory organs and nervous systems in these early fish, laying the groundwork for the later evolution of jaws. The Silurian also marked a crucial period for the evolution of corals and the expansion of reef ecosystems. While rudimentary reefs existed in the Ordovician, the Silurian saw the proliferation of large, complex reef structures built by tabulate and rugose corals, along with stromatoporoids, sponge-like organisms. These reefs were not just geological formations, they were vibrant, three-dimensional underwater cities that provided shelter, food, and breeding grounds for an immense variety of marine life. The creation of these extensive reef systems fundamentally altered ocean habitats, increasing ecological complexity and fostering biodiversity. Within these reefs, new species of snails, bivalves, and other invertebrates flourished, forming intricate food webs. The stability and complexity offered by these reefs were vital for the overall recovery and diversification of marine life. The changing global climate also played a role in the rebound. As the massive glaciers retreated and the Earth warmed, sea levels rose again, flooding continental shelves and creating vast, shallow, epicontinental seas. These warm, sunlit waters were ideal for the proliferation of marine life. The increased area of shallow water habitats provided ample space for new species to evolve and spread. The chemical composition of the oceans also stabilized, allowing for the widespread deposition of limestones, which are often rich in fossilized remains, testifying to the burgeoning life. The Silurian oceans were characterized by clear, warm waters, supporting a rich diversity of filter feeders, scavengers, and emerging predators. The recovery was not instantaneous. It was a gradual process spanning millions of years. The initial phase involved the survival of generalist species that could tolerate a wide range of environmental conditions. As conditions stabilized, these survivors began to specialize and diversify, leading to an increase in species richness. This period also saw the co-evolution of predator-prey relationships becoming more complex. While early predators like nautiloids continued to be important, the emergence of more agile and specialized fish and arthropods intensified the evolutionary arms race, driving further adaptations in both predators and their prey. The Silurian period therefore represents a critical chapter in the history of life, demonstrating how ecosystems can recover and even surpass their previous levels of diversity after a major environmental upheaval, setting the stage for the dramatic evolutionary leaps of the Devonian period. During the Silurian period, roughly 443 to 419 million years ago, and extending into the early Devonian, the shallow seas of Earth were dominated by a formidable group of arthropods known as eurypterids, commonly referred to as sea scorpions. These ancient creatures were not true scorpions, though they shared some superficial resemblances, particularly in their segmented bodies and often pincer-like appendages. Eurypterids ranged greatly in size and form, from small, agile swimmers to colossal apex predators, some of which grew to be larger than a human being. Their reign as the top predators in many marine and brackish water environments was a testament to their evolutionary success and their remarkable adaptations. One of the most impressive and well-known of the giant eurypterids was Pterygotus. This magnificent creature could reach lengths of over 2 meters, or approximately 6.5 feet, making it one of the largest arthropods to have ever lived. Imagine an animal of this size, with a broad, flattened head, powerful swimming paddles at the rear of its body, and large, crushing claws at the front. Pterygotus was a truly fearsome sight in the ancient seas. Its body was segmented and covered in a chitinous exoskeleton, which provided protection and support. The most striking features of Pterygotus were its massive chelicerae, the pincer-like appendages near its mouth. These were not delicate structures. They were robust, serrated claws capable of grasping and crushing prey, indicating a powerful predatory lifestyle. Its large compound eyes, positioned on the top of its head, suggest it was an active visual hunter, likely ambushing its prey from above or from the murky depths. The hunting strategies of these giant sea scorpions were likely varied, depending on their specific morphology and habitat. For Pterygotus, its large swimming paddles, formed from modified legs, would have allowed it to move with considerable speed and agility through the water, making it an effective pursuit predator. It probably hunted early fish, such as the armored jawless ostracoderms, as well as other large invertebrates like trilobites and cephalopods. The powerful crushing claws would have been ideal for breaking through the armor of its prey. Some eurypterids, particularly those with less developed swimming paddles and more robust walking legs, might have been ambush predators, lying in wait on the seafloor or in dense algal beds, striking out at unsuspecting victims. Others may have been scavengers, feeding on carrion that fell to the seabed. Eurypterids occupied a wide range of ecological niches. While some were clearly apex predators, others were smaller and likely fed on detritus or smaller invertebrates. Their success was partly due to their adaptability to various aquatic environments. While many were marine, inhabiting shallow coastal waters, some groups evolved to live in brackish estuaries and even freshwater lakes and rivers. This ability to colonize diverse habitats allowed them to spread widely and exploit different food resources. The presence of gills for breathing underwater, combined with a robust exoskeleton, made them well suited to these aquatic environments. Some species even show adaptations that suggest they might have been able to briefly venture onto land, perhaps for mating or to escape predators, though they remained fundamentally aquatic. The dominance of sea scorpions in the Silurian and early Devonian was also facilitated by the relative scarcity of other large, agile predators. While nautiloids were significant predators, they were generally slower and less maneuverable than the eurypterids. The early jawed fish were still relatively small and not yet as diverse or abundant as they would become later in the Devonian. This lack of direct competition from highly efficient vertebrate predators allowed eurypterids like Pterygotus to occupy the top rung of the food chain in many ecosystems. They were the undisputed kings of their watery domains, shaping the evolution of their prey through intense predatory pressure. However, the reign of the giant sea scorpions was not eternal. As the Devonian period progressed, jawed fish underwent a rapid and dramatic diversification, evolving into larger, faster, and more efficient predators. Placoderms, the armored jawed fish, and early sharks began to fill the apex predator niches. These new vertebrate predators, with their superior speed, maneuverability, and powerful jaws, outcompeted the eurypterids. While smaller eurypterids persisted into the Carboniferous and even the Permian, the colossal forms like Pterygotus declined as the Devonian age of fish truly took hold. The fossil record shows a clear shift in dominance from the arthropod giants to the vertebrate giants, marking a significant transition in marine ecosystems. Nevertheless, for millions of years, the sea scorpions, particularly the awe-inspiring Pterygotus, ruled the ancient seas, leaving behind a legacy as some of the most impressive predators in Earth's prehistoric oceans. The evolution of jaws stands as one of the most pivotal innovations in the history of life, fundamentally transforming marine ecosystems and paving the way for the incredible diversity of vertebrates we see today, including humans. Before the advent of jaws, the aquatic world was dominated by creatures that fed in simpler ways. Early filter feeders, like many of the Cambrian and Ordovician invertebrates, passively strained microscopic particles from the water. Others were scavengers, relying on dead organic matter, or detritivores, sifting through sediments. The earliest fish, the ostracoderms, were jawless. They feed by sucking in water and filtering out small organisms, or by scraping algae and detritus from surfaces using a muscular pharynx. While effective for their time, these feeding methods limited their dietary options and their ability to actively pursue and consume larger, more complex prey. The development of jaws opened up an entirely new dimension of predation and resource exploitation. The origin of jaws is a remarkable story of evolutionary repurposing. Scientists believe that jaws evolved from the gill arches, the skeletal supports that held the gills in jawless fish. In early vertebrates, these gill arches were positioned near the mouth and helped to pump water over the gills for respiration. Over time, the anterior, or front, gill arches became modified and enlarged, gradually shifting forward to form the upper and lower jaws. This transformation was not a sudden event, but a gradual process driven by natural selection, favoring individuals with increasingly effective biting mechanisms. The ability to articulate these modified arches to open and close them with force provided an unprecedented advantage. The immediate and profound impact of jaws was the revolution in predation. For the first time, vertebrates could actively grasp, bite, and chew their prey. This allowed them to capture larger, more active organisms that were previously inaccessible. Prey could be held firmly, torn apart, or crushed, leading to more efficient consumption of nutrients. This new predatory capability created an evolutionary arms race, driving the diversification of both predators and prey. Prey animals evolved stronger armor, faster escape mechanisms, and more complex behaviors to avoid being caught, while predators evolved more powerful jaws, sharper teeth, and improved sensory abilities to detect and capture their meals. This dynamic interaction accelerated the pace of evolution in marine environments. The earliest known jawed fish belonged to a group called the acanthodians, often referred to as spiny sharks due to their superficial resemblance to modern sharks and the prominent immovable spines that supported their fins. Acanthodians first appeared in the Silurian period approximately 430 million years ago and flourished throughout the Devonian before eventually declining in the Permian. They were generally small to medium-sized fish, typically ranging from a few centimeters to about 2 meters in length. Their bodies were slender and streamlined, suggesting they were active swimmers. Unlike modern fish, acanthodians possessed numerous small, bony scales covering their bodies, giving them a somewhat armored appearance. Acanthodians were characterized by their distinctive array of spines. Each of their fins, including the paired pectoral and pelvic fins, and often multiple dorsal and anal fins, was preceded by a stout, unjointed spine. These spines likely served as a defense mechanism against larger predators, making them difficult to swallow. Their jaws were simple but effective, often lined with small, sharp teeth, indicating a predatory diet. They likely fed on smaller invertebrates and other fish, using their speed and newly acquired biting ability to capture prey. Their presence in the fossil record, often in large numbers, suggests they were successful and widespread in both marine and freshwater environments. The development of jaws in acanthodians marked the divergence of the two major lineages of jawed fish that dominate aquatic ecosystems today. The chondrichthyans, cartilaginous fish, including sharks, rays, and chimaeras, and the osteichthyans, bony fish, which include ray-finned fish and lobe-finned fish. While the acanthodians are now considered a distinct extinct group, they represent the foundational step in the evolution of all subsequent jawed vertebrates. Their existence demonstrates that the basic blueprint for jawed feeding was established early in vertebrate history. The revolution brought about by jaws was not just about predation. It also allowed for new ways of processing food, leading to the exploitation of a wider range of food sources. Herbivory, crushing shells, and specialized feeding on particular types of organisms became possible. This increased dietary flexibility contributed to the explosion of fish diversity in the Devonian period, which is famously known as the Age of Fish. With jaws, fish could occupy virtually every ecological niche in the aquatic realm, from bottom-dwelling scavengers to fast-swimming open ocean predators. The evolutionary success of jawed fish dramatically reshaped marine food webs, leading to more complex and dynamic ecosystems than ever before. The legacy of this single evolutionary innovation continues to define the structure and function of aquatic life on Earth. The Paleozoic Era, a span of time stretching from about 541 million years ago to 252 million years ago, witnessed remarkable transformations in Earth's oceans. Among the most significant were the evolutionary innovations that led to the diverse forms of fish we see today. One such group, the placoderms, stands as a testament to early vertebrate experimentation, characterized by their distinct and often massive bony armor. Placoderms were a class of armored prehistoric fish, unique in their structure and their independent evolution of jaws. They first appeared in the Silurian period, around 440 million years ago, and thrived throughout the Devonian period before largely disappearing in the late Devonian extinction event about 360 million years ago. A few lingered into the Carboniferous period. Their name, placoderm, literally means plate-skinned, a direct reference to the thick, bony plates that covered their heads and often the front part of their bodies. This armor was not merely for protection, it was an integral part of their skeletal structure, forming a rigid shield around their vital organs. Unlike later fish, their internal skeletons were largely made of cartilage, which rarely fossilizes well. Thus, most of what scientists know about placoderms comes from these well-preserved bony plates, particularly their skulls and shoulder girdles. The armor was typically made of interlocking plates, providing a strong defense against predators. This defensive strategy was crucial in oceans that were becoming increasingly populated with large invertebrates and early predatory fish. Early placoderms, such as the antiarchs or the tiny Botryolepis, give us a glimpse into the initial stages of this armored lineage. These early forms were generally smaller than their more famous Devonian descendants like Dunkleosteus. Botryolepis, for instance, discovered in Australia, was a small, elongated placoderm with a unique, long snout, suggesting a specialized feeding strategy, perhaps for grubbing in soft sediments. Its small size, likely less than 30 centimeters, about 12 inches in length, meant its armor provided crucial protection in a world where even small creatures faced threats. These early placoderms often occupied bottom-dwelling niches, feeding on small invertebrates or detritus. Their relatively flat bodies and robust armor would have made them well-suited for a life close to the seafloor, providing stability and defense. A key evolutionary breakthrough for placoderms, and indeed for all jawed vertebrates, was the development of jaws. Before placoderms, most vertebrates, like the ostracoderms, were jawless filter feeders or detritivores. The evolution of jaws in placoderms was a significant independent event. Scientists believe that jaws evolved from the gill arches, the skeletal supports for the gills. In placoderms, the first gill arch evolved into the upper and lower jaws, while others transformed into supporting structures. This innovation allowed placoderms to actively bite, crush, and manipulate their food, opening up entirely new dietary possibilities and ecological roles. With jaws, they could become active predators, herbivores, or scavengers, a stark contrast to their jawless ancestors. This ability to bite was a game-changer, facilitating the radiation of placoderms into diverse forms and sizes. The structure of placoderm jaws was also distinctive. They did not have true teeth that were replaced throughout their lives like modern sharks or bony fish. Instead, their jaws often had sharp, bony plates or self-sharpening blades that protruded from the jawbone. In some cases, these plates were extensions of the jaw itself, creating scissor-like cutting edges. For example, Arthrodires, a group of placoderms that included Dunkleosteus, had sharp, bony gnathal plates that functioned like teeth. These plates were often deeply serrated or ridged, providing efficient cutting or crushing surfaces depending on their diet. This unique dental arrangement highlights an alternative evolutionary path to predation, compared to the development of replaceable teeth seen in other vertebrate lineages. The placoderm body plan was typically divided into two main sections, a head shield and a trunk shield, which were often connected by a movable joint. This joint allowed the head to articulate relative to the body, enabling a wider gape and more efficient feeding. This articulation was a specialized feature not seen in other armored fish groups. The trunk shield, while also armored, was often less extensively covered than the head, particularly in smaller, more agile forms. This varied level of armor suggests different lifestyle adaptations, with some placoderms being heavily defended bottom dwellers and others more streamlined for swimming in the open water. The tail section of placoderms was generally unarmored and provided propulsion, though its exact form varied among different groups. Placoderms filled a wide array of ecological niches in the Paleozoic oceans. While some were large, apex predators, many early and smaller forms were not. Some placoderms were likely bottom feeders, sifting through sediments for small invertebrates or organic matter. Their flattened bodies and strong head shields would have protected them while foraging on the seafloor. Others may have been omnivores, consuming both plant matter and small animals. The diversity of their jaw structures, ranging from sharp cutting edges to broad crushing plates, indicates a wide spectrum of diets across the group. For example, some placoderms possessed grinding plates, suggesting they fed on shellfish or other hard-bodied prey. The development of paired fins, including pectoral and pelvic fins, also allowed for greater maneuverability and control in the water, further enabling them to exploit different parts of the marine environment. These fins, while not as highly developed as in modern fish, still represented a significant evolutionary advance for controlled swimming. While oceans teemed with diverse and evolving life during the Paleozoic era, a quiet revolution was beginning along the shorelines, one that would profoundly reshape the entire planet, including its aquatic environments. For billions of years, land surfaces had been barren, exposed to the sun's harsh ultraviolet radiation and the forces of wind and erosion. Life, particularly complex life, was confined to the water. However, around 470 million years ago in the Ordovician period, the first simple plants began their hesitant colonization of dry land. This was not a sudden invasion, but a gradual process, marked by the evolution of specific adaptations that allowed organisms to survive outside the watery realm. The earliest land plants likely evolved from freshwater green algae. These algae possessed many of the pigments and reproductive strategies that would prove advantageous for life on land. However, moving from water to land presented immense challenges. Life in water provides constant support, easy access to water and dissolved nutrients, and a stable temperature. On land, organisms face desiccation, drying out, the need for structural support against gravity, a means to absorb water and nutrients from the soil, protection from ultraviolet radiation, and a way to reproduce without relying on water for sperm dispersal. The pioneering land plants were small, low-growing, and relatively simple. One of the most important and well-known early land plants is Cooksonia. Fossil evidence of Cooksonia has been found in various parts of the world, including Europe and North America, dating back to the late Silurian period, about 425 million years ago. Cooksonia was a tiny plant, typically only a few centimeters tall, resembling a small fork or Y-shaped stick. It consisted of simple, dichotomously branching stems, meaning they split into two equal parts that ended in spore-producing structures called sporangia. These sporangia were round or oval capsules where spores were produced and released into the air, allowing for dispersal by wind. Crucially, Cooksonia lacked true roots, stems, or leaves as we understand them today. Instead, it likely had a simple, root-like structure called a rhizoid that anchored it to the ground and absorbed some water and minerals. Its stems were probably photosynthetic, meaning they could produce their own food using sunlight. To combat desiccation, Cooksonia had a waxy cuticle, a protective outer layer that helped prevent water loss through evaporation. It also possessed stomata, small pores on its surface that could open and close to regulate gas exchange, taking in carbon dioxide and releasing oxygen while minimizing water loss. These were critical adaptations for survival in an aerial environment. The shift of plants to land was not merely an expansion of life's territory. It initiated a cascade of profound ecological changes. Initially, these small plants would have had a limited impact. However, as they diversified and spread across continents over millions of years, their collective presence began to alter the Earth's atmosphere, climate, and geology in fundamental ways. One of the most significant impacts was on the composition of the atmosphere. Plants take in carbon dioxide during photosynthesis and release oxygen. As land plants proliferated, they began to draw down vast amounts of atmospheric carbon dioxide, leading to a significant increase in oxygen levels over time. This increase in oxygen had far-reaching consequences for all life, including marine organisms, as it directly affected atmospheric and oceanic chemistry. The establishment of plant life on land also had a dramatic effect on soil formation and nutrient cycling. Prior to land plants, rock was directly exposed to weathering, leading to relatively coarse sediments entering waterways. Plants, with their rhizoids and eventually roots, began to break down rocks, creating finer soil particles. The decay of plant matter added organic material to the soil, further enriching it and creating a more complex substrate. This newly formed soil, rich in nutrients, was then subject to erosion by rain and rivers. This erosion carried increased amounts of dissolved nutrients, such as phosphorus and nitrogen, into rivers and eventually into the oceans. This influx of nutrients from land had a massive, though complex, impact on marine ecosystems. In small amounts, these nutrients could stimulate primary productivity in coastal waters, leading to localized blooms of algae and supporting a richer food web. However, in larger quantities, particularly after major periods of plant expansion, this nutrient runoff could lead to eutrophication. Eutrophication is the excessive richness of nutrients in a body of water, often due to runoff from the land, which causes a dense growth of plant life and death of animal life from lack of oxygen. Massive algal blooms could consume vast amounts of oxygen when they decompose, creating dead zones where most marine life could not survive. This phenomenon might have contributed to some of the smaller extinction events seen in the Paleozoic marine fossil record. Furthermore, the stabilization of soil by plant root systems reduced the amount of sediment washed into rivers and seas. While initially increased erosion from early plant colonization might have led to more sediment runoff over longer timescales, the establishment of stable plant communities would have reduced this, leading to clearer coastal waters in some areas. The development of vast, swampy forests, particularly during the Carboniferous period, also contributed to the burial of massive amounts of organic carbon, leading to the formation of coal deposits. This carbon sequestration further impacted atmospheric carbon dioxide levels and global climate. In essence, the colonization of land by simple plants like Cooksonia was not just a minor environmental shift, it was a planetary engineering project spanning millions of years. It fundamentally altered the Earth's atmosphere, soil, and nutrient cycles, creating a feedback loop that continually reshaped both terrestrial and marine environments. The oceans, while seemingly separate, were inextricably linked to these terrestrial changes, experiencing shifts in oxygen, nutrient availability, and sediment influx, profoundly influencing the evolution and distribution of marine life. The Devonian period, spanning from approximately 419 million years ago to 359 million years ago, is universally known as the Age of Fishes. This designation is not an exaggeration. During this time, fish underwent an explosive diversification, evolving into a bewildering array of forms that filled almost every available ecological niche in the vast oceans. It was a period of unprecedented innovation in fish evolution, laying the groundwork for many of the fish groups we see today. From the seafloor to the open water, fish dominated the marine realm, showcasing an incredible range of sizes, shapes, and lifestyles. One of the defining features of the Devonian was the continued success and radiation of placoderms, the armored fish discussed previously. While some, like the infamous Dunkleosteus, were immense apex predators, reaching lengths of up to 10 meters, about 33 feet, many other placoderm groups also thrived. Antiochs, for instance, were bottom-dwelling placoderms with highly specialized armored pectoral fins, allowing them to walk on the seabed or stir up sediment to find prey. Ptyctodonts were another group, often smaller and characterized by crushing tooth plates, suggesting a diet of shellfish. The diversity among placoderms alone demonstrates the varied roles fish were beginning to play in marine ecosystems. Alongside the placoderms, another significant group of armored fish, the ostracoderms, though beginning to decline, still persisted in the early Devonian. These jawless fish, with their external bony shields, represented an older lineage. However, their numbers and diversity were being overshadowed by the rise of jawed fish, which were simply more efficient and versatile predators and feeders. The evolution of jaws was a key factor in the Devonian fish explosion, allowing fish to exploit a wider range of food sources and establish complex food webs. Perhaps the most important evolutionary event of the Devonian was the rapid diversification of two major groups of bony fish, the ray-finned fish, Actinopterygii, and the lobe-finned fish, Sarcopterygii. These two groups represent the ancestors of nearly all living fish and all land vertebrates, respectively. Ray-finned fish, characterized by fins supported by bony rays, underwent massive radiation. Early forms were generally small, but they quickly diversified into various shapes, including swift swimmers and bottom dwellers. Their flexible fins allowed for highly maneuverable swimming, enabling them to navigate complex underwater environments and pursue prey effectively. This group would eventually come to dominate marine and freshwater environments after the Paleozoic Era. Lobe-finned fish were equally, if not more, significant from an evolutionary perspective. Their fins were fleshy and muscular, supported by a central bone structure resembling a limb. This unique fin structure allowed them to walk on the seafloor or move through shallow, oxygen-poor waters. The two primary groups of lobe-finned fish were the coelacanths and the lungfish, both of which have living descendants today. However, the most critical lineage of lobe-finned fish were the Rhipidistians, which included fish like Eusthenopteron. These fish possessed internal nostril openings and a primitive lung-like structure, enabling them to gulp air from the surface, a crucial adaptation for surviving in stagnant or low-oxygen waters. From one group of Rhipidistians, a lineage of fish known as the tetrapodomorphs evolved, giving rise to the first vertebrates with limbs, the tetrapods, which would eventually colonize land. This transition from fin to limb, while taking place in shallow aquatic environments, was a direct consequence of the unique fin structure of lobe-finned fish. The Devonian also saw the emergence and diversification of early sharks, chondric thighs. Unlike bony fish, sharks have skeletons made of cartilage, which rarely fossilizes well. However, their teeth and scales are often preserved, providing evidence of their increasing presence. Early sharks were not the massive predators we often imagine today, but they were efficient swimmers and predators, relying on their speed and sharp teeth. Cladocelac, a relatively small and streamlined shark from the late Devonian, is one of the best-known early shark forms. It possessed a unique fin structure and a powerful tail, indicating it was an active hunter in the open ocean. The presence of sharks added another layer of complexity to the Devonian marine food webs. The Devonian oceans were not uniform. There were deep oceanic basins, shallow coastal seas, and vast reef systems. Fish adapted to all these environments. Reefs, which had been growing in complexity since the Ordovician, reached immense sizes in the Devonian, particularly in what is now Western Australia. These reefs, built by corals and stromatoporoids, sponge-like organisms, provided diverse habitats, shelter, and abundant food sources, contributing to the high biodiversity of fish. Fish occupied every trophic level, from filter feeders sifting plankton from the water column to bottom dwellers scavenging detritus to mid-water hunters pursuing smaller fish and invertebrates and, of course, the apex predators that dominated the food chain. The sheer abundance and variety of fish forms in the Devonian reflect an era of intense evolutionary innovation and ecological specialization. The development of jaws, the diversification of fin structures, and the adaptation to a wide range of marine habitats allowed fish to become the dominant life forms in the aquatic realm. The Devonian period truly cemented the fish as kings of the water, a legacy that continues to this day. The Devonian period, spanning from about 419 million to 359 million years ago, is often called the Age of Fish. While many different kinds of fish thrived, one creature stood out as the undisputed king of the ancient seas, Dunkleosteus. This enormous placoderm, an armored fish, was one of the largest predators of its time, dominating the marine food webs of the late Devonian. Its name, Dunkleosteus, means Dunkel's bone, after David Dunkel, a curator at the Cleveland Museum of Natural History. Dunkleosteus was not a typical fish as we know them today. It belonged to a group called Arthrodires, which were armored placoderms. Instead of teeth, it possessed self-sharpening bony plates that formed razor-sharp edges capable of slicing through bone. These jaw plates were part of its skull and were incredibly strong. The front pair of these plates formed a beak-like structure, while another pair behind them created a crushing surface. When Dunkleosteus opened its mouth, these plates would slide past each other, creating a scissor-like action. When it closed its mouth, the front plates would interlock, providing a powerful grip. This unique dental arrangement allowed it to exert immense biting forces. Estimates suggest that Dunkleosteus could deliver a bite force of up to 6,000 newtons, or about 1,350 pounds per square inch, at the tip of its jaws. This was one of the most powerful bites of any animal, past or present, second only to some large crocodilians and Tyrannosaurus rex. This incredible bite force enabled Dunkleosteus to feed on other heavily armored fish and even early sharks, crushing their protective scales and bones. Its sheer size was a major factor in its predatory success. Fossil evidence, primarily from large, robust skull plates, suggests that Dunkleosteus could reach lengths of up to 10 meters, or about 33 feet. Some estimates even push this closer to 11 meters. Its body was heavily armored, particularly the head and the front part of its trunk, which were covered in thick bony plates. This armor provided excellent protection from rival predators, though Dunkleosteus itself had few, if any, natural enemies once it reached adult size. The rest of its body, including its tail, was likely unarmored, possibly covered in skin or scales, which would have allowed for more flexible movement in the water. The unarmored tail section, however, is rarely preserved in the fossil record, making its exact shape somewhat speculative, though reconstructions often depict it with a powerful eel-like or shark-like tail for propulsion. Dunkleosteus was a fast-moving apex predator. Its streamlined body shape, despite the heavy armor on its front, suggests it was an active hunter, not a sluggish bottom dweller. It likely patrolled the open waters of the Devonian seas, preying on whatever it could catch. Its diet would have included other placoderms, early sharks, and even large bony fish. Its massive size and powerful bite allowed it to capture and consume prey, much larger than most other predators. The discovery of partially digested fish remains within Dunkleosteus fossil deposits suggests it was a voracious eater. Some fossils have also shown evidence of bone trauma, indicating battles with other large marine creatures, possibly even other Dunkleosteus individuals, as they competed for food or territory. The habitat of Dunkleosteus was primarily marine, particularly in the shallow, warm seas that covered parts of North America, Europe, and North Africa during the Devonian period. Fossils have been found in various locations, with some of the most significant discoveries in the Cleveland Shale of Ohio, United States, where multiple well-preserved heads have been unearthed. These rich fossil beds provide detailed insights into the anatomy and lifestyle of this ancient predator. The presence of Dunkleosteus fossils alongside those of other large marine animals indicates a thriving and complex ecosystem where this placoderm occupied the highest trophic level. Its widespread distribution across different continents suggests it was a successful and adaptable species, able to thrive in various marine environments. Despite its dominance, Dunkleosteus eventually became extinct at the end of the Devonian period during a series of extinction events known as the Late Devonian Extinction. The exact reasons for its disappearance are not fully understood, but global climate changes, fluctuating sea levels, and a decrease in oxygen levels in the oceans are thought to have played a role. The extinction event significantly affected marine life, leading to the decline and disappearance of many groups, including the placoderms. While Dunkleosteus was a highly specialized and powerful predator, its adaptations might have made it vulnerable to rapid environmental shifts that favored more generalist species or those with different physiological requirements. Its legacy, however, remains as one of the most iconic and terrifying fish to have ever lived, a true monster of the ancient deep. Sharks, with their sleek bodies, rows of sharp teeth, and fearsome reputation, are among the most successful predators in modern oceans. However, their lineage stretches back hundreds of millions of years, long before the dinosaurs. The earliest true sharks emerged during the Devonian period from about 419 million to 359 million years ago, making them one of the oldest groups of vertebrates still alive today. These early sharks were quite different from their modern counterparts, representing a foundational stage in their evolutionary history. One of the best-known examples of these primordial sharks is Cladocelache. Cladocelache, meaning branch shark, lived during the late Devonian period, about 370 million years ago. Its fossils are remarkably well preserved, particularly in the Cleveland Shale of Ohio, United States, where the fine sediments help to preserve not only its skeleton, but also impressions of soft tissues, such as muscle fibers and even kidney traces. This exceptional preservation provides a rare glimpse into the anatomy of these early cartilaginous fish. Unlike the bony fish that were diversifying rapidly at the time, sharks are cartilaginous, meaning their skeletons are made of cartilage rather than bone. While cartilage rarely fossilizes as well as bone, the unique conditions of these ancient seabeds allowed for Cladocelache's delicate skeletal structure to be preserved. One of the most striking differences between Cladocelache and modern sharks was its body shape and fin structure. Cladocelache was relatively small compared to later sharks, typically reaching lengths of up to 1.8 meters or about 6 feet. Its body was torpedo-shaped, clearly adapted for fast swimming. However, its fins were quite stiff and broad, particularly the paired pectoral and pelvic fins. These fins lacked the flexibility seen in modern sharks, which use their fins for precise maneuvering. Instead, Cladocelache's fins were more like fixed wings, providing lift and stability, similar to how an airplane's wings function. This suggests that Cladocelache was a swift swimmer that relied on bursts of speed to catch prey rather than intricate turns and maneuvers. Its caudal, or tail fin, was homocercal, meaning its upper and lower lobes were roughly equal in size, a feature not commonly seen in modern sharks, which usually have a heterocercal tail with a longer upper lobe. The jaw structure of Cladocelache also differed significantly from modern sharks. Its jaws were firmly attached to its skull, a condition known as amphistole. Modern sharks have jaws that are more loosely connected to the skull, allowing them to protrude their mouths forward to seize prey, a highly effective feeding adaptation. Cladosolach's fixed jaw limited its biting range, but still allowed it to effectively capture small, fast-moving prey. Its teeth were multi-cusped, meaning they had several points and were relatively small, triangular, and pointed. They were not shed and replaced continuously in rows like those of modern sharks, but rather remained in place. This indicates a different feeding strategy, likely involving seizing and swallowing prey whole or in large chunks rather than tearing flesh. Fossilized gut contents have revealed that Cladosolach fed on small bony fish, crustaceans, and other marine invertebrates, suggesting it was an opportunistic predator in the Devonian ecosystem. Another notable feature of Cladosolach was the absence of scales on most of its body. Unlike modern sharks, which are covered in dermal denticles, tiny tooth-like scales that reduce drag and offer protection, Cladosolach had a largely smooth skin. Only around its mouth and on its fins were some denticles present. This smooth skin might have further contributed to its streamlined profile and speed. It also possessed two dorsal fins, each preceded by a spine, a feature seen in some modern sharks as well. The presence of these spines may have offered some defense against larger predators like Dunkleosteus, though adult Cladosolach were likely prey to the largest placoderms. The habitat of Cladosolach was marine, primarily in the open waters of the Devonian seas. Its adaptations for speed suggest it was an active hunter in the pelagic zone, the open water column, rather than a bottom dweller. It coexisted with a wide array of other marine life, including diverse bony fish, trilobites, and the formidable placoderms. Cladosolach's success demonstrates the early evolutionary potential of cartilaginous fish and their ability to carve out a predatory niche in a world dominated by armored forms. Its appearance marks a crucial step in the long and successful history of sharks, establishing many of the fundamental characteristics that would be refined and diversified over subsequent geological periods, leading to the diverse array of sharks we see today. While Cladosolach itself did not survive past the Devonian extinction event, its anatomical features provide a direct link to the earliest forms of these ancient ocean hunters. The journey of life from water to land is one of the most profound evolutionary transitions in Earth's history. A critical step in this journey involved a group of fish known as the Sarcopterygians, or lobe-finned fish. Unlike the more common ray-finned fish, actinopterygians, whose fins are supported by slender, bony rays, lobe-finned fish possess fleshy, muscular fins supported by a central array of robust bones. This unique fin structure was not merely an adaptation for swimming, but a crucial pre-adaptation that laid the groundwork for the evolution of limbs in land-dwelling vertebrates. The earliest lobe-finned fish appeared in the late Silurian period, about 420 million years ago, and diversified significantly during the Devonian period. The defining characteristic of lobe-finned fish is their fins. Each paired fin, whether pectoral, front, or pelvic rear, contains a single bone articulating with the shoulder or hip girdle, followed by a series of other robust bones that resemble the limb bones of land animals. These bones are surrounded by a thick layer of muscle and flesh, giving the fin a lobed appearance, distinct from the fan-like structure of ray-finned fish fins. This internal skeletal support allowed lobe-finned fish to use their fins not just for propulsion through water, but also for supporting their body weight and rudimentary movement on solid surfaces, such as the bottom of shallow waters, or even temporarily on land. This ability to walk or prop themselves up was a key innovation. Early lobe-finned fish were predominantly aquatic. They lived in a variety of freshwater and marine environments. For example, Primitive forms like Eusthenopteron, which lived about 380 million years ago in the late Devonian, possessed strong, bony fins and likely inhabited shallow, oxygen-poor freshwater environments. The robust nature of their fins would have allowed them to maneuver through dense aquatic vegetation or even to prop themselves up in stagnant pools. Another important group was the lungfish, Dipnoi, which are still alive today, represented by species found in Africa, South America, and Australia. Modern lungfish have a unique adaptation. They possess lungs in addition to gills, allowing them to breathe air when water levels drop or oxygen becomes scarce. This dual breathing ability was likely present in many ancient lobe-finned fish and would have been highly advantageous in environments prone to drying out. The coelacanths, Actinistia, represent another ancient lineage of lobe-finned fish, famously thought to have gone extinct at the end of the Cretaceous period, until a living specimen was discovered off the coast of South Africa in 1938. Modern coelacanths live in deep marine environments and use their lobed fins for slow, deliberate movements, almost like walking on the sea floor. Their discovery provided invaluable insight into the soft tissue anatomy and movement patterns of these ancient fish. While they are not direct ancestors of land vertebrates, their existence highlights the diversity and longevity of the lobe-finned fish lineage. The most crucial group of lobe-finned fish in the context of the water-to-land transition are the rhipidistians, specifically the tetrapodomorphs. These fish, which include famous fossils like Panderichthys and Tiktaalik, show a progressive series of adaptations towards terrestrial life. Panderichthys, from the late Devonian, had a flattened head, eyes on top of its head, and a more robust skeleton, suggesting it lived in very shallow water, perhaps lying in wait for prey. Its paired fins showed further development of internal bone structures that increasingly resembled the limb bones of land animals. Tiktaalik rosé, discovered in 2004, is often hailed as a missing link because it beautifully bridges the gap between fish and four-legged land animals, tetrapods. Living about 375 million years ago, Tiktaalik possessed a unique blend of fish and tetrapod features. Like a fish, it had scales, fins, and gills. However, it also had a flattened skull with eyes on top, a mobile neck, and most importantly, robust fin skeletons with bones that clearly correspond to the humerus radius and ulna of a tetrapod limb, as well as a primitive wrist. These strong, flexible fins could have allowed Tiktaalik to prop itself up in very shallow water, or even move across muddy substrates in a push-up motion. The presence of a rib cage that could support its body weight on land and evidence of functional lungs further solidified its transitional status. The evolution of these walking fins was not an immediate leap onto dry land, but rather a gradual process of adapting to life in shallow, complex aquatic environments, such as swamps, deltas, and tidal flats. In these environments, strong fins capable of propelling or supporting the body through obstacles or over short distances on exposed land would have offered advantages for escaping predators, finding new food sources, or moving between drying pools. The fleshy, bony fins of sarcopterygians provided the raw material, the anatomical blueprint for the subsequent evolution of the full-fledged limbs of the first amphibians, marking an extraordinary chapter in the story of life on Earth. Life on Earth began in the water, and for billions of years, it stayed there. The oceans teemed with creatures, from simple single-celled organisms to complex armored fish. But around 375 million years ago, a remarkable transition began. A lineage of fish, known as lobe-finned fish, started to take the first tentative steps towards a life on land. This was not a sudden leap, but a slow, gradual adaptation driven by changing environments. The world during the Middle and Late Devonian period was different from today. Continents were still shifting, and vast, shallow, warm seas covered much of the land. These shallow waters often led to extensive swampy areas choked with the first simple land plants like ferns and mosses. As these plants grew, they began to change the landscape and, importantly, the water. Decaying plant matter would have enriched the water with nutrients, leading to bursts of algae growth. This dense vegetation and decaying matter would have depleted oxygen in the water, creating challenging conditions for typical fish that relied on gills for breathing. Lobe-finned fish possessed unique features that gave them an advantage in these oxygen-poor, swampy habitats. Unlike ray-finned fish, whose fins are supported by bony rays, lobe-finned fish had fleshy, muscular fins with a central bone structure. This bone structure was strikingly similar to the bones found in the limbs of all land animals, or tetrapods. These sturdy fins were not just for swimming, they could also be used to push off the muddy bottom, to navigate through dense underwater vegetation, or even to lift the head out of the water. One of the most crucial adaptations was the presence of a primitive lung. Most fish breathe using gills, extracting oxygen from the water. However, some early lobe-finned fish, like modern lungfish, developed a simple sac connected to their digestive tract that could absorb oxygen directly from the air. This was incredibly valuable in stagnant, deoxygenated waters. If the water became too low in oxygen, these fish could simply gulp air from the surface. This dual breathing system meant they were less dependent on the water's oxygen levels and could survive in places where other fish would perish. Consider a creature like Eusthenopteron, an early lobe-finned fish that lived around 385 million years ago. While still very much a fish, its fin bones showed the basic pattern that would later evolve into the humerus, radius, and ulna of a limb. It was an active predator in Devonian lakes and rivers, using its strong fins to maneuver in complex underwater environments. It represents an early stage of the skeletal changes that allowed for terrestrial movement. The fossil record reveals a series of transitional forms, each showing slightly more advanced adaptations for a semi-aquatic existence. One of the most famous and important of these is Tiktaalik rosei, discovered in the Canadian Arctic in 2004. Tiktaalik lived approximately 375 million years ago. It is often described as a fishapod because it possessed a mosaic of fish and tetrapod characteristics. Like a fish, Tiktaalik had scales, fins, and gills. However, its head was flattened, more crocodile-like, with eyes on top, suggesting it spent time looking upwards out of the water. Crucially, its fins were even more robust than those of Eusthenopteron. The bone structure within Tiktaalik's fins was complete, showing a clear shoulder, elbow, and wrist joint, making them strong enough to support its body weight. This meant Tiktaalik could likely prop itself up in shallow water and perhaps even push up across muddy banks or submerged logs. The wrist bones in Tiktaalik's fins were particularly significant. They allowed for flexibility at the end of the limb, a key feature for maneuvering on solid ground. Its ribs were also larger and more overlapping than those of typical fish, providing better support for its internal organs outside the buoyancy of water. Furthermore, Tiktaalik possessed a mobile neck, meaning its head could move independently of its shoulders, unlike most fish. This would have been useful for looking around in shallow water or on land, perhaps to spot prey or avoid predators. The development of these features in Tiktaalik was not for extensive land travel. Its environment was probably very shallow, murky water, full of obstacles like fallen logs and dense vegetation. In such a place, a creature that could push itself through tight spots, prop its head out of the water to gulp air, or even crawl short distances from one puddle to another, would have a significant advantage in finding food or escaping larger aquatic predators. It was about moving within the swamp, not necessarily abandoning water entirely. Following Tiktaalik in the fossil record, we find creatures like Acanthostega, which lived around 365 million years ago. These were definite tetrapods, possessing clear limbs with digits rather than fin rays. Acanthostega had 8 digits on each limb and still spent most of its life in water, though its limbs show adaptations for navigating vegetated aquatic environments. Ichthyostega was even more robust, with 7 digits on its hind limbs, larger ribs, and a stronger backbone, indicating it was better suited for supporting its weight on land, though it too remained largely aquatic. The shift from fins to limbs was a monumental event in Earth's history. It allowed vertebrates to escape the confines of the ocean and eventually colonize every terrestrial habitat. The strong, muscular fins of lobe-finned fish, combined with their ability to breathe air, were the initial steps. The swampy, oxygen-poor environments of the Devonian acted as a natural selective force, favoring those individuals with features that allowed them to survive in such challenging conditions. This slow, incremental process, spanning millions of years, represents one of the most profound evolutionary transitions, ultimately leading to the diversity of life on land we see today. The Paleozoic era was punctuated by several major extinction events. But the late Devonian mass extinction, often referred to as the Second Great Dying, stands out for its prolonged and devastating impact on marine life. This was not a single, sudden event, but rather a series of pulses that unfolded over several million years, roughly between 372 and 359 million years ago. While not as famous as the Permian-Triassic extinction, which marked the end of the Paleozoic, the late Devonian crisis profoundly reshaped ocean ecosystems and wiped out a significant portion of marine biodiversity. The most severe phase of this extinction is known as the Kellwasser event, occurring approximately 372 million years ago. During this time, a massive decline in marine species, particularly those living in shallow, warm waters, became evident in the fossil record. The impact was global, affecting oceans across the supercontinent Laurussia and the vast Panthalassic Ocean. Unlike some other mass extinctions linked to clear asteroid impacts or massive volcanic eruptions, the precise causes of the late Devonian extinction are still debated among scientists. However, a leading hypothesis points to widespread oceanic anoxia, which means a severe lack of oxygen in the ocean waters. This anoxia would have rendered vast areas of the seafloor uninhabitable for most complex life forms. The evidence for this comes from black shales, rich in organic carbon and depleted in oxygen-sensitive minerals, found in late Devonian rock formations across the globe. These black shales indicate periods where the deep ocean water became stagnant and oxygen-poor. What could have caused such widespread anoxia? One compelling theory involves the rapid expansion of land plants during the Devonian. Plants like ferns, lycopods, and early trees were colonizing land at an unprecedented rate, forming the first true forests. While a monumental step for terrestrial life, this growth had significant consequences for the oceans. As plants grew, their roots broke down rocks and soils, releasing nutrients like phosphorus and nitrogen into rivers. These rivers then carried the nutrient-rich runoff into the shallow coastal seas. An influx of nutrients into the ocean can lead to massive algal blooms, a process known as eutrophication. When these immense algal mats eventually died, they sank to the seafloor. The decomposition of this vast amount of organic matter by bacteria consumed huge quantities of oxygen in the water. In stagnant, poorly mixed ocean basins, this consumption could lead to anoxic conditions, particularly in the deeper waters. The development of deeper root systems in plants also led to increased chemical weathering of rocks, further enhancing nutrient runoff. Another contributing factor could have been climate change. Evidence suggests a cooling trend during the late Devonian, possibly linked to the widespread growth of land plants. As forests expanded, they drew down significant amounts of carbon dioxide from the atmosphere through photosynthesis. carbon dioxide is a greenhouse gas. So, a reduction in its levels could have led to global cooling and potentially even glaciations. Cooler temperatures can reduce ocean circulation, further exacerbating anoxia as cold water holds more dissolved gases, but stratified layers can prevent oxygen from reaching deeper parts of the ocean. Glaciation would also have led to falling sea levels, reducing the amount of habitable shallow water shelf environments. The late Devonian extinction disproportionately affected marine life. Reef-building organisms were hit particularly hard. The once thriving coral stromatoporoid reef systems, which had dominated the shallow, tropical seas for millions of years, almost entirely collapsed. These complex ecosystems, teeming with diverse life, largely disappeared, and reefs would not fully recover their previous diversity until much later in the Mesozoic era. Among the specific groups that suffered severe losses were the armored fish, the placoderms. These large, dominant predators, like Dunkleosteus, were greatly reduced in number and diversity, and most groups eventually went extinct by the end of the Devonian. Trilobites, which had been incredibly diverse since the Cambrian, also experienced significant declines. Many of their families and genera disappeared, though some hardy lineages survived into the Carboniferous. Brachiopods, sessile filter feeders, and crinoids, often called sea lilies, also saw major reductions in their diversity. While fish generally suffered, the impact was not uniform. Lobe-finned fish and early ray-finned fish, perhaps due to their adaptability to varying oxygen levels and their more generalized diets, were less affected than the specialized placoderms. This relative survival of early bony fish and amphibians proved crucial, as these groups would go on to dominate the aquatic and terrestrial realms in later periods. The exact mechanisms and triggers of the late Devonian extinction are still under investigation. It was likely a complex interplay of environmental changes. The rise of land plants, while a biological triumph, inadvertently triggered a cascade of events in the oceans. Increased nutrient runoff, leading to algal blooms, subsequent widespread anoxia, and potentially global cooling. This period serves as a stark reminder of the interconnectedness of Earth's systems and how biological innovations on one part of the planet can have profound and unexpected consequences elsewhere. The marine world that emerged from the Devonian extinction was fundamentally altered, paving the way for new evolutionary trajectories in the Carboniferous period. Following the late Devonian mass extinction, Earth entered the Carboniferous period, which spanned from approximately 359 to 299 million years ago. This era is famously characterized by its vast, lush and humid terrestrial environments dominated by enormous coal-forming swamps. These swamps were not just a feature of the land. Their pervasive influence profoundly shaped the adjacent marine environments, leading to a world quite different from the one that existed before. The continents during the Carboniferous were still in motion. Gondwana, a supercontinent, was drifting northward, and it began to collide with Euramerica, forming the new supercontinent of Pangaea in the later parts of the period. This continental arrangement, combined with a generally warm and humid global climate, created ideal conditions for the explosive growth of plants. Equatorial regions, in particular, were home to extensive land forests. These forests were unique. They were not dominated by flowering plants or modern conifers, which had not yet evolved. Instead, the landscape was covered by giant lycopod trees like Lepidodendron and Sigillaria, towering ferns and horsetails, many reaching heights of 30 meters or more. These plants thrived in perpetually wet, marshy conditions, forming dense, dark and swampy forests. When these plants died, they fell into the anoxic, oxygen-poor water of the swamps. Due to the lack of oxygen, the organic matter did not fully decay. Instead, it slowly accumulated, compacting over millions of years to form the enormous coal deposits that give the Carboniferous period its name. The sheer scale of these terrestrial swamps had direct and indirect effects on the marine environment. Rivers flowing from these heavily vegetated landmasses carried vast amounts of sediment and organic matter into coastal waters. This continuous influx created wide, shallow, muddy marine shelves and extensive deltas. These environments were often turbid, with low light penetration due to suspended particles, and they could experience fluctuating salinity and oxygen levels, especially in estuaries and lagoons directly connected to the swampy rivers. The decay of immense quantities of plant material in the swamps also affected atmospheric composition. Photosynthesis by the vast forests drew down enormous amounts of carbon dioxide from the atmosphere, leading to a significant increase in atmospheric oxygen levels. Oxygen levels during the late Carboniferous are estimated to have reached 30 to 35%, significantly higher than the 21% we experience today. This high oxygen level had implications for both terrestrial and marine life. On land, it allowed for the evolution of giant arthropods, like dragonflies with wingspans of over 2 feet. In the oceans, while not as directly impactful as terrestrial changes, stable oxygen availability in open marine environments was generally favorable for many forms of marine life. However, the localized conditions in coastal marine environments adjacent to the swamps were different. The runoff from the swamps, laden with organic material and nutrients, likely led to periods of localized algal blooms and subsequent anoxia in restricted bays and lagoons, similar to what may have occurred during the Devonian. These areas would have supported specialized communities adapted to low oxygen and high organic content. Despite these localized challenges, the open oceans of the Carboniferous continued to host diverse marine life. Following the late Devonian extinction, new groups began to diversify. Sharks, which had originated in the Devonian, experienced major radiation. The Carboniferous is sometimes called the Age of Sharks due to their incredible diversity and abundance. Many unusual forms evolved, including the spiral-toothed helicoprion, the anvil-headed stethocanthus, and the strange simorium. These sharks occupied a wide range of ecological niches, from coastal predators to open ocean cruisers. Ray-finned fish also continued their diversification. While less visually striking than the placoderms that dominated the Devonian, these early bony fish were increasingly agile and adaptable, laying the groundwork for the vast diversity of modern fish. They began to fill the ecological roles left vacant by the extinction of many placoderms. The great coral reef systems of the Devonian had largely collapsed during the extinction, and while some reef-like structures began to re-emerge in the Carboniferous, they were generally less extensive and diverse than their Devonian predecessors. Bryozoans, crinoids, and sponges contributed to the formation of smaller, patchier bioherms rather than massive continuous reefs. Ammonoids, a group of shelled cephalopods related to modern nautiluses and squids, flourished in the Carboniferous seas. They developed complex suture lines on their shells, a feature used by paleontologists to identify different species and stages of evolution. They were mobile predators and scavengers, common in both shallow and deeper waters. Bivalves and gastropods, types of mollusks, also became more common and diverse, burrowing in the seafloor or grazing on algae. Sea scorpions, or eurypterids, while past their Silurian peak, still persisted in coastal and brackish waters, including some of the swampy estuarine environments. Some species were quite large, remaining important predators in these transitional habitats. In essence, the Carboniferous marine world was a blend of established groups recovering from extinction and new groups diversifying. The overwhelming presence of terrestrial coal swamps profoundly influenced the coastal and shallow marine zones, creating environments rich in organic matter and sometimes low in oxygen. These unique conditions fostered specialized ecosystems. While in the more open oceans, fish and sharks continued their evolutionary journey, paving the way for the marine life of the Permian period. The Paleozoic Era, a vast stretch of geological time spanning hundreds of millions of years, witnessed an astonishing diversification of life, particularly within its expansive oceans. Among the most remarkable and visually striking inhabitants of these ancient seas were the crinoids, often referred to as sea lilies due to their plant-like appearance. These fascinating creatures, though superficially resembling plants, were in fact animals belonging to the phylum Echinodermata, the same group that includes modern-day starfish, sea urchins, and brittle stars. Their explosion in numbers and diversity led to the formation of immense underwater landscapes known as crinoid meadows, vast living carpets that covered significant portions of the seafloor for millions of years. Crinoids first appeared in the early Ordovician period, approximately 485 million years ago. But it was during the Silurian and Devonian periods, and especially into the Carboniferous period, that they truly flourished. Their basic body plan consisted of a holdfast, which anchored the animal to the seafloor, a stalk composed of numerous disc-shaped ossicles or plates, a calyx or body cup at the top of the stalk containing the mouth and other organs, and a crown of feathery branching arms. These arms, sometimes numbering in the hundreds, were covered in tiny tube feet and served as efficient filter-feeding structures. The crinoid would extend its arms into the water column, passively capturing microscopic plankton and detritus drifting by. This filter-feeding strategy was highly successful in the nutrient-rich waters of the Paleozoic. The formation of crinoid meadows was a testament to their incredible abundance. Imagine an ancient seafloor bathed in clear, warm, relatively shallow waters, perhaps a few tens of meters deep. Instead of sandy plains or rocky outcrops, vast areas would have been dominated by dense stands of crinoids, swaying gently with the ocean currents, much like terrestrial meadows of grass or fields of flowers. These meadows could extend for many square kilometers, creating a unique, three-dimensional habitat that provided shelter and food for countless other marine organisms. The stalks of individual crinoids varied in length from a few centimeters to several meters, allowing them to reach different levels within the water column, maximizing their feeding efficiency and creating a layered ecosystem. The sheer biomass of these crinoid forests was immense, representing a significant component of the Paleozoic marine food web. Within these bustling crinoid meadows, a complex ecosystem thrived. The crinoids themselves were primary consumers, converting the energy from microscopic organisms into their own body mass. This made them a crucial food source for various predators. Early jawed fish, such as the placoderms and early sharks, would have navigated through the crinoid stalks, preying on smaller fish, invertebrates, and perhaps even the crinoids themselves. Though the crinoids' hard, calcareous plates offered some protection, nautiloids, large-shelled cephalopods, and eurypterids, or sea scorpions, also hunted in these environments. The spaces between the crinoid stalks provided shelter for a myriad of smaller creatures. Brachiopods, which were bivalve-like filter feeders, often attached themselves to crinoid stalks or the seafloor. Bryozoans, tiny colonial animals that formed encrusting mats, and various worms and other soft-bodied invertebrates found refuge and food among the crinoid forest. Scavengers would have fed on detritus and the remains of dead organisms that settled on the seafloor. The crinoid meadows played a vital role in shaping the marine environment. Their holdfasts helped to stabilize the seafloor sediment, preventing erosion and creating more stable habitats for other benthic organisms. Their continuous filter feeding helped to clarify the water, influencing light penetration and nutrient cycling. Furthermore, upon their death, the calcareous plates and ossicles of crinoids contributed significantly to the formation of vast limestone deposits. Many of the limestone formations we see today, particularly those from the Carboniferous period, are largely composed of fragmented crinoid remains, offering a direct fossil record of their immense past abundance. These fossilized remains are so common that geologists can often identify ancient seafloors by the presence of crinoid fossils. While the stalked crinoids were the most prominent in these meadows, some crinoids were unstalked and able to move freely across the seafloor or even swim short distances. These free-swimming forms became more prevalent in later geological periods, but the vast majority of Paleozoic crinoids were sessile, anchored to the substrate. The success of crinoids was due to their adaptability and their efficient filter-feeding mechanism, allowing them to colonize a wide range of marine environments, from shallow shelves to deeper waters. Though the densest meadows were typically found in well-lit, shallow and calm conditions, the reign of the crinoid meadows continued largely uninterrupted until the end of the Paleozoic Era. The Permian-Triassic extinction event, the most severe mass extinction in Earth's history, devastated marine life, including the crinoids. While many lineages perished, some crinoids survived this cataclysm. Today, crinoids still exist in modern oceans, primarily in deep-sea environments, where they are often referred to as living fossils. However, they no longer form the expansive, dense meadows that characterized the Paleozoic seas. The scale and ecological dominance of the ancient crinoid meadows remain a unique feature of Earth's deep past, a testament to a time when filter-feeding echinoderms created living landscapes on an epic scale, shaping marine ecosystems for hundreds of millions of years. Their story highlights the dynamic nature of life on Earth and the incredible variety of forms that have emerged and flourished in the planet's long history. The Devonian period, often called the Age of Fish, ended with a series of extinction events that significantly impacted marine life, particularly the dominant armored fish known as placoderms. These formidable predators, such as the massive Dunkleosteus, had ruled the Devonian seas, but their decline left significant ecological niches open. It was into this newly available space that sharks, a group of cartilaginous fish, began to truly diversify and establish their long-standing dominance as apex predators. While early shark-like forms appeared in the Silurian, their true takeover of marine ecosystems began in earnest during the Carboniferous period, following the placoderms' demise. Sharks possess several key adaptations that contributed to their evolutionary success. Unlike most fish, their skeletons are made of cartilage rather than bone. While this means their fossil record is often limited to teeth and fin spines, it also provides a flexible, lightweight, and strong framework. They typically have multiple gill slits, powerful jaws lined with rows of continuously replaced teeth, and a streamlined body shape optimized for efficient swimming. These features allowed them to become highly effective hunters in various marine environments. As the Carboniferous period unfolded, approximately 359 million years ago, the oceans were ripe for new predators, and sharks were perfectly positioned to fill that role. The Carboniferous and Permian periods saw an astonishing radiation of shark forms, some of which were truly bizarre and unlike any sharks alive today. These ancient sharks experimented with a wide array of body plans and feeding strategies, showcasing the incredible evolutionary plasticity of the group. Two of the most famous examples of these unusual ancient sharks are Stethocanthus and Helicoprion, each possessing unique and perplexing anatomical features. Stethocanthus, a genus of shark that lived from the late Devonian to the early Carboniferous, approximately 370 to 320 million years ago, was a relatively small shark, typically growing to about 1 to 2 meters in length. Its most striking feature was its dorsal fin complex. Instead of a typical triangular fin, Stethecanthus possessed an anvil-shaped or ironing board-shaped structure on its back, covered in a brush-like patch of enlarged denticles, which are tiny tooth-like scales. Curiously, a similar patch of denticles was also found on the top of the male Stethecanthus's head. The function of this peculiar structure has been a subject of much scientific debate. Several hypotheses have been proposed for the purpose of Stethecanthus's anvil-shaped fin. One popular theory suggests it was involved in mating rituals, perhaps acting as a display to attract mates or to help males grip females during copulation. Another idea is that it served a defensive purpose, making the shark appear larger or more intimidating to potential predators. Some researchers have speculated it might have played a role in attracting prey, perhaps mimicking a larger fish or using the denticles to create a turbulent flow that disoriented small prey. It is also possible that it served a combination of these functions. Regardless of its exact purpose, the unique anatomy of Stethecanthus highlights the diverse evolutionary paths taken by early sharks. These sharks inhabited shallow coastal waters, likely preying on smaller fish and invertebrates. Their fossil remains have been found in North America and Europe, providing valuable insights into early shark evolution. Even more enigmatic was Helicoprion, a shark that lived from the late Carboniferous to the early Triassic, approximately 310 to 250 million years ago. Helicoprion was a large shark, with estimates suggesting it could reach lengths of up to 7 or 8 meters, making it one of the largest predators of its time. Its most distinctive feature was an extraordinary tooth whorl, or spiral of teeth, that was embedded in its lower jaw. This spiral structure contained all the teeth the shark had ever produced, with older, smaller teeth at the center of the spiral and newer, larger teeth at the outer edge. For decades, the exact placement and function of Helicoprion's tooth whorl were a mystery. Early reconstructions placed the spiral on the dorsal fin, on the tail, or even in the throat. However, more recent research, particularly through the use of computer tomography, CT scans of fossilized skulls, has confirmed that the tooth whorl was indeed located within the lower jaw, extending backwards into the mouth cavity. The function of this unique dental apparatus is still debated, but leading theories suggest it acted like a circular saw, rotating to slice through prey, or perhaps was used to crush soft-bodied animals. One prominent hypothesis is that Helicoprion preyed on soft-bodied cephalopods, like ammonites, using the tooth whorl to cut through their shells or to extract them from their shells. It might also have fed on fish. The continuous growth and replacement of teeth within the spiral would have ensured a constantly sharp and effective feeding tool. Helicoprion inhabited deep ocean environments, suggesting it was a pelagic predator. Its survival through the Permian-Triassic extinction event, though it eventually died out in the early Triassic, underscores its evolutionary resilience. Beyond Stethecanthus and Helicoprion, other ancient sharks also exhibited unusual forms. Xenacanthus, for instance, was a freshwater shark with an eel-like body and a distinctive spine projecting from the back of its head. Orthocanthus, another freshwater genus, possessed a double-spined dorsal fin. These diverse forms illustrate that the Carboniferous and Permian were periods of great experimentation for sharks as they adapted to various ecological niches and environments, from shallow coastal waters to deep oceans and even freshwater systems. The success of sharks in the wake of the placoderm extinction was profound. Their cartilaginous skeletons, efficient predatory adaptations, and continuous tooth replacement system allowed them to thrive and diversify. While many of the bizarre forms of the Paleozoic eventually died out, the fundamental body plan of sharks proved incredibly robust, enabling them to survive multiple mass extinction events, including the devastating Permian-Triassic extinction. Their lineage continues to this day, with modern sharks representing the culmination of hundreds of millions of years of evolutionary refinement, a testament to the shark takeover that began in the ancient seas of the Carboniferous. The transition of life from water to land is one of the most profound evolutionary events in Earth's history. While the earliest forms of life originated and diversified in the oceans, the Carboniferous period, approximately 359 to 299 million years ago, marked a pivotal time when vertebrates truly began to conquer terrestrial environments. This era is often referred to as the Age of Amphibians, due to the remarkable diversification and ecological dominance of these creatures in the vast, humid swamp forests and freshwater systems that characterized the period. These large predatory amphibians represented the first major group of vertebrates to thrive both in and out of the water, showcasing a critical step in the journey of life onto land. The ancestors of amphibians were lobe-finned fish, a group known as Sarcopterygi, which possessed fleshy, muscular fins, supported by bones that were homologous to the limb bones of land vertebrates. Throughout the Devonian period, approximately 419 to 359 million years ago, a series of transitional forms emerged, illustrating the gradual evolution of features necessary for terrestrial life. Key fossil discoveries like Tiktaalik, often called a fishapod, showed a creature with fish-like scales and fins, yet possessing a flattened skull, eyes on top of its head and a flexible neck, along with robust fin skeletons that hinted at weight-bearing capabilities. Later forms, such as Ichthyostega and Acanthostega, were more clearly tetrapods, meaning they had 4 limbs. While still highly aquatic, with gills and fin rays in their tails, they also possessed lungs and limbs capable of limited movement on land, particularly in shallow, oxygen-poor waters or for short excursions between ponds. By the Carboniferous, the descendants of these early tetrapods had diversified into a wide array of amphibians. The global climate during this period was warm and humid, leading to the formation of extensive swamp forests, particularly in the equatorial regions. These coal forests, named for the vast coal deposits they formed from decaying plant matter, provided ideal habitats for amphibians: abundant freshwater, lush vegetation offering shelter and food, and a rich invertebrate prey base including giant insects. The amphibians of this era were not the small, smooth-skinned creatures we typically associate with frogs and salamanders today. Many were large, formidable predators, well adapted to their semi-aquatic lifestyles. The characteristics of these early amphibians reflected their transitional nature. They possessed lungs for breathing air, a crucial adaptation for life out of water. But many still retained gills or relied on cutaneous respiration, breathing through their skin, which necessitated keeping their skin moist. Their limbs, while stronger and more articulated than fish fins, were often sprawling, making locomotion on land somewhat cumbersome, especially for larger species. Consequently, most Carboniferous amphibians remained closely tied to water, spending much of their lives in rivers, lakes, and swamps, returning to water for reproduction. Their eggs lacked a hard shell and would quickly dry out on land, requiring them to be laid in water, where the larval stage, tadpole-like, would develop before metamorphosing into the adult form. Among the most iconic of the large predatory Carboniferous amphibians was Eriops. This robust amphibian, which lived during the early Permian but evolved from Carboniferous ancestors, could grow up to 2 meters, about 6.5 feet in length. It had a broad, flattened skull with powerful jaws and sharp teeth, suggesting a diet of fish, smaller amphibians, and possibly early reptiles. Eriops possessed strong, well-developed limbs that allowed it to move effectively on land for short periods, likely to bask in the sun or move between water bodies. But it was primarily a semi-aquatic predator, lurking in the shallow waters of swamps and ponds. Its heavily built skeleton and large size made it a dominant predator in its environment. Another fascinating amphibian was Diplocalus, famous for its boomerang-shaped head. This distinctive skull, formed by elongated bones extending from the back of its head, gave it a unique appearance. Diplocalus was smaller than Eryops, typically around 1 meter, about 3 feet long, and was more fully aquatic. The function of its boomerang head is debated. Theories include its use as a hydrofoil to help it swim efficiently, a defensive mechanism to make it difficult for predators to swallow, or even as a way to trap prey. Its flattened body and relatively weak limbs suggest it was not well suited for terrestrial movement, spending its life in freshwater environments, likely preying on fish and invertebrates. Even larger amphibians existed, such as Proterogerynus, a giant temnospondyl amphibian from the Permian of Brazil. This crocodile-like creature could reach lengths of up to 9 meters, nearly 30 feet, making it the largest amphibian known to have ever lived. Its long, narrow snout, filled with sharp teeth, indicates it was a powerful aquatic predator, similar in lifestyle to modern crocodiles, preying on large fish and other aquatic vertebrates in freshwater systems. Mastodonsaurus, another massive amphibian from the Triassic, also had Carboniferous ancestors and could reach 6 meters in length, with an enormous skull, reinforcing the image of these early amphibians as formidable predators. The rise of amphibians marked a significant ecological shift. They became the dominant vertebrate predators in freshwater and semi-aquatic environments, filling niches that had previously been occupied by large fish. Their ability to move onto land, even if only for short periods, opened up entirely new food resources and escape routes from aquatic predators. This dual existence, thriving both in and out of the water, demonstrated the incredible adaptability of early tetrapods and laid the groundwork for the evolution of truly terrestrial vertebrates. However, despite their success, amphibians faced inherent limitations due to their reliance on water for reproduction and their permeable skin. As the climate began to dry towards the end of the Carboniferous and into the Permian, and as reptiles, with their amniotic eggs and scaly desiccation-resistant skin, evolved and diversified, amphibians faced increasing competition. The Permian-Triassic extinction event delivered a final blow to many amphibian lineages, though some groups survived and continued to evolve, eventually leading to modern amphibians. The Carboniferous amphibians, with their diverse and often bizarre forms, stand as a powerful testament to a time when life was truly thriving in the liminal zone between water and land, paving the way for all future terrestrial vertebrate life. The ancient world of the Carboniferous and early Permian periods, spanning roughly from 359 million to 252 million years ago, was a time of remarkable biological gigantism, particularly among insects. While our focus is primarily on marine creatures, it is important to acknowledge the interconnectedness of all ecosystems. Freshwater environments teeming with life flowed into the vast prehistoric seas, and the creatures inhabiting these rivers and lakes were part of the broader Paleozoic tapestry. Among the most iconic inhabitants of these freshwater systems were the colossal insects, whose sheer size remains a subject of fascination. This era was characterized by exceptionally high atmospheric oxygen levels, which played a crucial role in enabling insects to grow to sizes unheard of today. The oxygen content in the air during the Carboniferous, for instance, is estimated to have been as high as 35%, significantly higher than the 21% we experience now. This abundance of oxygen directly impacted the respiratory systems of insects. Unlike vertebrates, insects do not have lungs that actively pump air. Instead, they rely on a passive system of tubes called tracheae, which branch throughout their bodies, delivering oxygen directly to their tissues. The efficiency of this tracheal system, and thus the maximum size an insect can attain, is limited by the diffusion rate of oxygen. With more oxygen in the atmosphere, oxygen could diffuse more effectively through these tracheal tubes, allowing for larger body sizes before oxygen supply became a limiting factor. The most famous example of this Paleozoic gigantism is Meganeura, a massive dragonfly-like insect that soared through the Carboniferous skies. With a wingspan that could reach up to 75 centimeters, or about 30 inches, Meganeura was truly a titan of the air, dwarfing any flying insect alive today. Its fossil remains, primarily found in France, provide clear evidence of its immense proportions. Meganeura was a formidable predator, much like modern dragonflies, but on a grander scale. It would have hunted other large insects, such as giant cockroaches and early forms of winged insects, as well as small amphibians that ventured too close to the water's edge. Its large, multifaceted eyes would have given it excellent vision for tracking prey, and its powerful veined wings allowed for swift, agile flight. The adult Meganeura was an aerial hunter, but its life began in the water. Like modern dragonflies, Meganeura had an aquatic larval stage, known as a nymph. These nymphs were also predatory, living in the freshwater swamps, lakes, and rivers that crisscrossed the Carboniferous supercontinent. They would have ambushed smaller aquatic invertebrates and perhaps even tiny fish or amphibian larvae, using specialized mouthparts to capture their prey. These aquatic stages highlight the direct link between the freshwater ecosystems and the broader Paleozoic environment. The health and productivity of these freshwater bodies directly influenced the success of these giant insects. Beyond Meganeura, other groups of insects also exhibited impressive sizes. For example, some ancient mayflies also had substantial wingspans, though not quite as large as Meganeura. While less is known about the aquatic larval stages of all these giant insects, it is generally understood that many Carboniferous insect orders had aquatic nymphs or larvae, playing important roles in the freshwater food webs. These included early forms of stoneflies and caddisflies, whose modern descendants still have aquatic larval stages. The presence of such large aquatic insect larvae would have significantly impacted the freshwater food chains, serving as both predators and prey for early fish and amphibians. The rivers and lakes of the Carboniferous and Permian were not isolated systems. They were integral parts of a global hydrological cycle, constantly feeding into the vast Panthalassa Ocean. Estuaries, where freshwater met saltwater, would have been dynamic zones of interaction. Though the giant insects themselves were primarily freshwater inhabitants, their existence underscores the rich biodiversity of the Paleozoic, extending beyond the purely marine realm. The lush, swampy forests of the Carboniferous, often referred to as the coal forests, provided the ideal habitat for these giant insects. These dense forests of lycopods, horsetails, and ferns created a warm, humid environment with abundant food sources and sheltered breeding grounds. The decay of this massive plant matter eventually formed the vast coal deposits that give the Carboniferous period its name. The high oxygen levels that permitted insect gigantism also fueled these immense forests, creating a positive feedback loop where more plants produced more oxygen, further supporting larger insect forms. The evolution of insect wings during the Devonian period had already opened up a new ecological niche, allowing insects to exploit aerial resources and escape ground-based predators. The Carboniferous then saw an explosion in winged insect diversity and, crucially, size. The decline of insect gigantism began towards the end of the Permian period and continued into the Mesozoic era. This reduction in size is largely attributed to a decrease in atmospheric oxygen levels, which began to fall from their Carboniferous peaks. As oxygen concentrations dropped, the physical limitations of the tracheal respiratory system once again became a dominant factor, favoring smaller body sizes. Additionally, the rise of new, more efficient aerial predators, such as early flying reptiles, pterosaurs, in the Mesozoic, may have also put evolutionary pressure on large insects, making smaller, more agile forms more advantageous for survival. The fossil record of these giant insects provides invaluable insights into the environmental conditions and evolutionary pressures of the Paleozoic era. Studying their morphology and ecological roles helps scientists reconstruct the complex food webs and environmental dynamics of a world vastly different from our own. The sheer scale of Meganeura and its contemporaries serves as a powerful reminder of life's capacity for adaptation and diversification under unique planetary conditions. Their aquatic larval stages in particular highlight the profound influence of freshwater ecosystems on the broader biological landscape of the ancient Earth, demonstrating that even creatures primarily known for their aerial prowess had crucial ties to the aquatic world. The interconnectedness of these ancient environments meant that changes in one system, such as the oxygen levels in the atmosphere, could have profound effects on the life cycles and maximum sizes of creatures living in both freshwater and indirectly marine environments. The Permian period, spanning from approximately 299 million years ago to 252 million years ago, marks the final chapter of the Paleozoic era. This geological epoch was a time of immense global change, primarily driven by the relentless forces of plate tectonics, which culminated in the assembly of the supercontinent Pangaea. The formation of this single colossal landmass profoundly reshaped Earth's geography, climate, and consequently its ocean currents, setting the stage for one of the most dramatic shifts in the history of life. Throughout the Carboniferous period, the Earth's continents had been slowly drifting towards each other. By the Permian, this convergence accelerated, leading to massive continental collisions. The ancient landmasses of Laurussia, North America and Europe, and Gondwana, South America, Africa, Antarctica, Australia, and India, finally merged, along with Siberia and other smaller continental blocks, to form Pangaea. This process was not a gentle coming together, but involved monumental geological events, including the uplift of vast mountain ranges, such as the ancestral Appalachian Mountains in North America and the Ural Mountains in present-day Russia, which formed as continents crumpled against each other. The sheer scale of Pangaea was unprecedented in Earth's history, covering nearly 1/3 of the planet's surface. The existence of such a vast landmass had a profound impact on global climate patterns. With most of the continental crust consolidated into a single body, the moderating influence of oceans was greatly diminished across much of the interior. This led to extreme continentality, characterized by vast deserts and arid conditions far from any coastline. Seasonal temperature fluctuations would have been much more pronounced than today, with scorching hot summers and bitterly cold winters across large swathes of the supercontinent. The formation of Pangaea also influenced global atmospheric circulation, leading to the development of powerful monsoonal climate systems that brought seasonal rains to some coastal regions, but left the interior parched. These drastic climatic shifts affected terrestrial ecosystems, leading to the decline of the Carboniferous coal swamps and the rise of more drought-tolerant plant species, such as conifers. Even more significantly for our understanding of ancient marine life, the formation of Pangaea drastically altered global ocean currents. Instead of several distinct ocean basins separated by continents, there was now a single, immense global ocean surrounding Pangaea, which paleogeographers refer to as Panthalassa. This superocean was vast and deep, stretching from pole to pole. The new configuration of land masses created entirely new patterns of ocean circulation. Without continents to act as barriers and direct currents, Panthalassa developed a relatively simple but powerful global current system. Large, slow-moving oceanic supergyres would have dominated the circulation, with warm equatorial waters flowing poleward and cooler waters returning towards the equator. The implications of these altered ocean currents were far-reaching for marine life. The restricted flow of water in certain areas, particularly in semi-enclosed basins or along the margins of Pangaea, could lead to oceanic stagnation and the formation of widespread anoxic zones. Anoxic conditions, where oxygen levels in the water are extremely low or absent, are detrimental to most marine life as they prevent respiration and the decomposition of organic matter. This could have led to marine dead zones where only specialized anaerobic bacteria could survive. Furthermore, the altered circulation patterns would have affected the distribution of nutrients throughout the ocean. Nutrient upwelling, which occurs when deep, nutrient-rich waters rise to the surface, is crucial for supporting marine productivity and the base of the food web. Changes in these upwelling patterns could have led to areas of both increased and decreased productivity, fundamentally reshaping marine ecosystems. The sheer size of Panthalassa also meant that its central regions were likely oligotrophic, meaning nutrient-poor, similar to the vast gyres of today's Pacific Ocean. Without the constant input of nutrients from continental runoff or extensive shallow water upwelling, these areas would have supported relatively sparse life. Coastal environments, particularly the shallow marine shelves, were also significantly impacted. As continents merged, the total length of coastline and the area of shallow shelf seas, which are typically the most biologically productive parts of the ocean, decreased. This reduction in habitable space put additional pressure on marine species that relied on these environments for breeding, feeding, and shelter. Despite these growing environmental stresses, marine life in the early to mid-Permian was still diverse. Brachiopods, sessile filter feeders resembling clams, continued to be abundant, as did crinoids, or sea lilies, which formed vast underwater meadows. Ammonoids, shelled cephalopods, related to modern squids and octopuses, were also diversifying, though their greatest radiation would occur in the Mesozoic. Fusulinids, a type of large, single-celled foraminifera, were particularly characteristic of Permian, shallow marine environments, forming significant components of the sediment. However, as the Permian progressed, the cumulative effects of Pangaea's formation, changing ocean chemistry, and fluctuating climates began to take their toll. The stage was being set for the most catastrophic event in Earth's history, an event that would bring the Paleozoic Era to a dramatic and definitive close, fundamentally reshaping the trajectory of life on Earth. The Permian period, therefore, serves as a crucial bridge, illustrating how geological forces can drive profound and lasting changes in both terrestrial and marine environments, ultimately leading to global biotic crises. For over 270 million years, from their first appearance in the early Cambrian period to their final disappearance at the end of the Permian trilobites, were among the most successful and diverse groups of marine arthropods to ever inhabit Earth's oceans. Their reign spanned nearly the entire Paleozoic era, during which they evolved into thousands of species, occupying a vast array of ecological niches. Their distinctive, three-lobed, segmented bodies, protected by a hard, mineralized exoskeleton, made them instantly recognizable in the fossil record. Trilobites were incredibly adaptable, living as bottom dwellers, filter feeders, scavengers, and even some possibly as active predators. They burrowed into soft sediments, crawled across the seafloor, and some even swam through the water column. Their compound eyes, often remarkably preserved, show a sophisticated visual system, with some species possessing unique schizochroal eyes that provide exceptional depth perception. However, despite their long and successful history, the story of trilobites is also one of gradual decline. punctuated by major extinction events, culminating in their final demise. Their first significant setback occurred at the end of the Ordovician period, around 443 million years ago, during a major global cooling event that led to widespread glaciation and a drop in sea levels. While many trilobite species perished, the group as a whole recovered and continued to diversify into the Silurian and early Devonian periods. Another major blow came during the late Devonian extinction event around 372 million years ago, which severely reduced their diversity. By the Carboniferous period, their numbers and morphological variety were greatly diminished. The once ubiquitous and diverse trilobites were now represented by only a few surviving orders, primarily the Proetida. In the Permian period, the final act of the trilobite saga unfolded. By this time, only a handful of trilobite species remained, clinging to existence in a world that was rapidly changing. These last trilobites were generally smaller and less ornate than their flamboyant Cambrian and Ordovician ancestors. They often lacked the elaborate spines, exaggerated eyes, or complex cephalic head structures seen in earlier forms. Instead, they were typically more streamlined and generalized, suggesting an adaptation to more marginal or less competitive ecological niches. The Pretida, the last surviving order, were typically small, smooth-shelled forms, often found in deeper water or in environments that were less impacted by the intense competition and environmental pressures of the shallow marine shelves. Their survival into the Permian was a testament to their resilience, but also highlighted their increasingly precarious position. Several environmental pressures contributed to the long, slow decline of the trilobites. One significant factor was the rise of new and more efficient predators. Throughout the Devonian period, jawed fish, including the formidable placoderms like Dunkleosteus and the first true sharks, underwent a massive evolutionary radiation. These new predators were faster, more agile, and possessed powerful jaws capable of crushing the trilobites' once impenetrable exoskeletons. While trilobites had evolved various defensive strategies, such as rolling into a ball or developing spines, these defenses became less effective against the increasingly sophisticated predatory techniques of the new fish. Habitat loss and change also played a crucial role as the supercontinent Pangaea assembled during the Permian. Global sea levels fluctuated, and the total area of shallow marine shelf environments, which were prime trilobite habitats, decreased. The formation of Pangaea also altered global ocean currents, leading to the development of widespread anoxic zones in the deep ocean, which were inhospitable to most marine life, including trilobites. Climate change, with cycles of warming and cooling, also contributed to environmental instability, further stressing populations already in decline. The relatively fixed body plan of trilobites, while successful for millions of years, may have limited their adaptability compared to more rapidly evolving groups like fish and mollusks. They lacked the internal skeletal flexibility that allowed vertebrates to diversify into new forms and ecological roles more readily. The fossil record of these last Permian trilobites is sparse, reflecting their reduced numbers and limited distribution. They're often found in specific geological formations, providing clues about their final refugia. Their extinction at the end of the Permian period, approximately 252 million years ago, was not a sudden event in isolation, but the culmination of a long decline, exacerbated by the most severe mass extinction event in Earth's history, the Permian Triassic extinction. This catastrophic event, which wiped out over 90% of marine species, delivered the final blow to the few remaining trilobite lineages. After a remarkable 270 million year reign, witnessing the rise and fall of countless other life forms, the trilobites, once the undisputed kings of the ancient seas, finally vanished from the planet, leaving behind an extraordinary fossil legacy that continues to inform our understanding of early life on Earth. Their long journey from the dawn of complex life to their ultimate disappearance serves as a powerful testament to the dynamic and ever-changing nature of our planet's biodiversity. The late Paleozoic era saw the emergence of a fascinating group of creatures, the synapsids, often referred to as mammal-like reptiles. Among them, some forms showed adaptations that hinted at a partial return to aquatic or semi-aquatic lifestyles, especially those with distinctive sails on their backs. While none were fully marine in the way later reptiles like ichthyosaurs or plesiosaurs would be, their presence in coastal or swampy environments during the Permian period marked a significant evolutionary step. These were not true reptiles in the strict modern sense, but a separate branch of amniotes, distinct from the lineage that would lead to dinosaurs and modern reptiles. The most notable of these sail-backed synapsids was Edaphosaurus. Edaphosaurus was a large, herbivorous animal that lived during the late Carboniferous and early Permian periods, roughly 300 million to 280 million years ago. Its most striking feature was a tall sail on its back, supported by long bony spines extending from its vertebrae. Unlike its more famous contemporary, Dimetrodon, which was a carnivore, Edaphosaurus possessed a different skull structure and teeth adapted for grinding plant matter. Its blunt, peg-like teeth were positioned in a crushing battery, suitable for processing tough vegetation. This suggests a diet primarily composed of plants. The environment in which Edaphosaurus lived was often characterized by extensive coal swamps and coastal lands. These were not open oceanic environments, but were instead dominated by ferns, horsetails, lycopods, and early conifers. The presence of Edaphosaurus fossils in such swampy or deltaic deposits suggests that it spent considerable time in or near water. While it lacked the specialized flippers or streamlined bodies of later marine reptiles, its general build was robust, capable of navigating muddy terrain and possibly wading in shallow water. Its relatively short limbs and broad body indicate it was not built for swift terrestrial pursuit, but rather for a more ponderous movement. The function of the dorsal sail in Edaphosaurus has been a subject of much scientific discussion. Several hypotheses exist, none definitively proven. One prominent idea is thermoregulation. The sail, being highly vascularized, meaning it had many blood vessels, could have absorbed solar heat quickly when oriented towards the sun, helping to warm the large body of the animal. Conversely, it could have dissipated excess heat when angled away from the sun or exposed to a breeze, preventing overheating. This would have been particularly advantageous for a large, slow-moving animal living in environments with fluctuating temperatures. Another hypothesis suggests the sail played a role in display, either for attracting mates or for intimidating rivals or predators. A large, brightly colored sail could have served as a visual signal in a dense, swampy environment. A third idea proposes it acted as a form of structural support or even as an anchor for powerful muscles, though this is less commonly accepted for Edaphosaurus compared to its carnivorous cousin. The presence of Edaphosaurus along coastlines and within swampy areas is significant because it represents an early, albeit indirect, connection between terrestrial amniotes and aquatic environments. While not a true marine reptile, its habitat choices and adaptations show how some lineages were exploring the edges of the water, a trend that would fully develop in the Mesozoic era with the emergence of specialized marine reptiles. These early synapsids, though part of a different evolutionary path than the reptiles that would dominate the Mesozoic seas, illustrate the consistent evolutionary pressure for terrestrial animals to return to the rich resources of aquatic environments. The move from land back to water is a recurring theme in evolutionary history, driven by factors such as abundant food sources, reduced competition, or escape from terrestrial predators. The fossil record of Edaphosaurus is found primarily in North America, particularly in what is now the Midwestern and Southwestern United States, as well as in Europe. These regions were part of the supercontinent Pangaea during the Permian. The consistent association of its fossils with freshwater or brackish water sediments rather than deep marine deposits confirms its semi-aquatic or coastal preference. Its diet of terrestrial plants further solidifies its ties to land-based vegetation, even if it spent considerable time in wet areas. The large size of Edaphosaurus, reaching lengths of up to 3.5 meters and weighing several hundred kilograms, made it a significant herbivore in its ecosystem. Its slow-moving nature meant it would have needed a stable, abundant food supply, which the extensive Permian swamps provided. The thick vegetation of these areas offered both sustenance and potential refuge. The sail-backed synapsids, including Edaphosaurus, eventually declined towards the end of the Permian period. Their ecological niche was eventually filled by other groups of animals. Their story, however, is crucial for understanding the broader tapestry of Paleozoic life. They demonstrate the diverse forms that evolved during this era and the varied strategies animals employed to thrive in their respective environments. The adaptations seen in Edaphosaurus, particularly its semi-aquatic leanings and its large size, were part of a broader trend of increasing complexity and specialization in Permian ecosystems. These animals, while perhaps not as iconic as the dinosaurs that followed, were vital components of their world, bridging the gap between purely terrestrial life and the subsequent extensive return to the seas by other reptilian lineages. Edaphosaurus represents a powerful example of evolutionary exploration at the interface of land and water, hinting at the vast, empty seas that would soon await new inhabitants after the Great Extinction. The Permian Triassic extinction event, often referred to as the Great Dying, stands as the most catastrophic biotic crisis in Earth's history. Approximately 252 million years ago, this event led to the demise of an estimated 96% of all marine species and 70% of terrestrial vertebrate species. It marked the end of the Paleozoic Era and ushered in the Mesozoic. The scale of this extinction dwarfs all others, including the one that ended the reign of the dinosaurs. Understanding its causes is crucial to comprehending the resilience and vulnerability of life on Earth. Scientific consensus points to massive volcanic eruptions in the Siberian Traps, a large igneous province located in modern-day Russia, as the primary trigger for this global catastrophe. These eruptions were not isolated events, but an extended period of intense volcanism that lasted for tens of thousands to hundreds of thousands of years. They released enormous quantities of volcanic gases, including carbon dioxide, methane, and sulfur dioxide, into the atmosphere. The sheer volume of these gases initiated a cascade of environmental changes that ultimately proved devastating to life. The immediate effect of massive carbon dioxide release was a significant increase in global temperatures, leading to extreme global warming. Models suggest average global temperatures could have risen by as much as 10 degrees Celsius. This warming had several detrimental consequences for marine life. Warming oceans led to a reduction in oxygen levels, a process known as ocean anoxia. Warmer water holds less dissolved gas, and the accelerated decomposition of organic matter in warmer conditions further consumes available oxygen. This created vast dead zones in the oceans where most complex life could not survive. Evidence for widespread anoxia is found in the geological record, with black, organic-rich shales deposited across ocean basins during this period. In addition to warming, the release of sulfur dioxide led to a period of initial acid rain. While this might have temporarily cooled the planet by forming aerosols, The long-term effect of massive carbon and methane emissions overwhelmed any cooling. More critically, carbon dioxide dissolved in ocean water led to severe ocean acidification. As the pH of the oceans dropped, marine organisms that built shells or skeletons out of calcium carbonate, such as corals, brachiopods, and many mollusks, found it increasingly difficult, if not impossible, to precipitate their hard parts. This severely disrupted marine food webs, as these shell-forming organisms formed the base for many ecosystems. Many marine invertebrate groups that had thrived throughout the Paleozoic, such as trilobites, blastoids, and many crinoid species, were completely wiped out. The release of methane, a potent greenhouse gas, from methane clathrates, frozen methane hydrates on the sea floor, also contributed to the extreme warming. The initial warming from carbon dioxide emissions could have destabilized these clathrates, leading to a runaway greenhouse effect that further accelerated global temperature rise and ocean anoxia. This positive feedback loop would have exacerbated the environmental stress on marine and terrestrial ecosystems. On land, the intense warming caused widespread aridity and desertification. The massive volcanic eruptions themselves also released toxic compounds that directly affected terrestrial environments. The combination of heat, lack of water, and potentially toxic air created an uninhabitable environment for many land plants and animals. The fossil record shows a dramatic loss of plant diversity and a collapse of complex forest ecosystems. This in turn deprived terrestrial herbivores of their food source, leading to a ripple effect throughout the food chain and devastating terrestrial vertebrate populations. The Permian-Triassic extinction was unique not just in its scale, but also in its duration. The environmental stresses persisted for an extended period, preventing quick recovery and pushing ecosystems past a tipping point. The oceans became stagnant, deoxygenated and acidic, hostile to most complex life. The land became a vast, arid wasteland. It was a time of extreme environmental upheaval, far beyond the capacity of most life forms to adapt or survive. Evidence for these events comes from various geological and paleontological studies. Detailed analyses of rock layers from the Permian-Triassic boundary show distinct geochemical anomalies, including spikes in carbon isotopes, indicating massive carbon releases into the atmosphere and oceans. Anoxic markers are also found in these layers. The fossil record clearly documents the dramatic disappearance of countless species across virtually all taxonomic groups at this boundary. For example, 80% of insect orders went extinct, highlighting the pervasive nature of the crisis across both marine and terrestrial realms. The Great Dying served as a reset button for life on Earth. It fundamentally reshaped the course of evolution, ending the dominance of Paleozoic fauna and paving the way for the rise of new groups in the Mesozoic. The recovery period was exceptionally long, lasting millions of years, underscoring the severity of the environmental changes. This event highlights the profound impact that large-scale geological processes can have on the biosphere, demonstrating Earth's capacity for self-destruction on a planetary scale. It stands as a stark reminder of the delicate balance that sustains life and the potential consequences of rapid, sustained environmental disruption. The immediate aftermath of the Permian-Triassic extinction event presented a chilling vision, a world ocean stripped of nearly all its complex life. The Great Dying had decimated marine ecosystems, leaving behind what scientists describe as the empty sea. With up to 96% of all marine species vanished, the oceans of the early Triassic period, approximately 251 million years ago, were eerily silent and profoundly altered. This stark emptiness marked the definitive end of the Paleozoic era and set the stage for a new chapter in life's history, Before the extinction, Paleozoic oceans teemed with diverse life. Vast coral algal reefs provided complex habitats for brachiopods, crinoids, bryozoans, and a multitude of mollusk species. Trilobites scuttled across the seafloor, while ancient fish and cephalopods navigated the water column. This rich tapestry of life, built over hundreds of millions of years, collapsed in a geologically instantaneous event. What remained was a radically simplified marine environment, almost devoid of large, complex organisms. The ecological structure of the oceans was fundamentally shattered. The reef-building organisms, which formed the very foundation of shallow marine ecosystems, were almost entirely wiped out. Calcareous sponges, rugose and tabulate corals, dominant reef builders of the Paleozoic, disappeared. The absence of these architects meant that the complex, three-dimensional habitats they created vanished, removing shelter and feeding grounds for countless other species. Instead, microbial mats, simple bacterial and algal communities became widespread across the seafloor. These mats, which thrive in oxygen-depleted and disturbed environments, covered vast areas, indicating stagnant and anoxic conditions that persisted long after the main extinction pulse. The water column itself was largely empty. The vast schools of Paleozoic fish were gone. Most cephalopods, including nautiloids, suffered massive losses, though some lineages, like the ammonoids, managed to survive in severely reduced numbers. Planktonic communities, the microscopic base of the marine food web, were also devastated, suggesting a widespread collapse of primary productivity. This further starved any remaining larger organisms. The overall biomass in the oceans plummeted to unprecedented lows. This period of emptiness, known as the Strangelove Ocean by some scientists, was characterized by sluggish ocean circulation and widespread anoxia, particularly in deeper waters. The warming of the oceans had reduced oxygen solubility, while massive input of nutrients from land, perhaps due to erosion in a barren world, fueled microbial blooms that consumed even more oxygen as they decomposed. The acidified conditions, resulting from volcanic carbon dioxide dissolving in seawater, continued to hinder the recovery of shell-building organisms. Surviving species were often small, opportunistic and generalist, capable of tolerating extreme conditions. The recovery of marine ecosystems after the Permian-Triassic extinction was protracted, taking millions of years. This long recovery period, sometimes referred to as the Triassic Gap or dead zone, is a stark indicator of the severity of the environmental trauma. In typical extinction events, recovery begins relatively quickly, within hundreds of thousands of years. However, after the Great Dying, the persistent hostile conditions, high temperatures, low oxygen, and ocean acidification inhibited the re-establishment of complex food webs and diverse communities. It took until the Middle Triassic, roughly 5 million to 10 million years after the event, for marine diversity to begin a substantial rebound. The types of organisms that eventually repopulated the oceans were often from lineages that had been minor players in the Paleozoic. For instance, new groups of reef builders, such as scleractinian corals, the ancestors of most modern corals, emerged. While ammonoids saw a resurgence, their diversity patterns were different. Most significantly, the empty ecological niches created by the extinction allowed for the diversification of entirely new forms of marine life. This ecological vacuum paved the way for the explosive radiation of new groups, particularly reptiles. With the oceans largely free of the dominant predators and competitors of the Paleozoic terrestrial reptile lineages, began to explore the aquatic realm, eventually leading to the evolution of highly specialized marine reptiles like ichthyosaurs, plesiosaurs, and mosasaurs in the subsequent Mesozoic era. The demise of ancient fish and cephalopods opened up predatory niches that these new marine reptiles would fill with remarkable efficiency. The empty sea of the early Triassic was not just a desolate void. It was a blank slate upon which the next chapter of marine evolution would be written. Driven by the unprecedented opportunities for innovation and diversification, the Paleozoic was truly over, and a new world was beginning to emerge from its ashes.